Welcome everyone. Today we are going to start a new chapter which is in 11th NCERT, chapter number 2, biological classification. So I already wrote few points on the board. Let us see that and then further we'll continue. Now the chapter name is biological classification. So in the first chapter in the living world we studied the significance of classification. 1.7 to 1.8 million species are there so far which are recorded. So in our lifetime it is not possible to study those 1.7 or 1.8 million species. So if we classify them under one phylum, if you study one species, almost all the species in that phylum will have the similar characteristics. Now from the time of dawn of civilization, so we tried to classify organisms. That's what they tell. Since the dawn of civilization, there has been many attempts to classify living organisms. Now the initial classifications are not scientific classifications. They are in fact made on the basis of whether they are useful organisms or they are not useful organisms. So that's what the author has mentioned in NCERT, that it was done instinctively, means without conscious thoughts, without any scientific basis. They have just checked whether the organisms are useful, they are needed or they are not needed. On that basis only they have done the initial classification. The first classification, it is Aristotle has done the earliest classification, which is more scientific we can tell, because he has classified plants into 3 groups called herbs, shrubs and trees, and he has also classified animals into 2 groups. The animals which are not having RBC are called Anima, and the animals which have RBC are called Inima. So on the basis of RBC, he divided the animals into 2 groups: without RBC, Anima; with RBC, Inima. Basing on the size, he has classified the plants into 2 groups, 3 groups. The first one is herbs, very short plants; shrubs, which are bushy; trees, which are very tall. So this was the first earliest classification, which is scientific classification. Then comes 2 kingdom classification, which is done by Carolus Linnaeus, right? A Swedish naturalist. His name is Carolus Linnaeus. We heard about Carolus Linnaeus, his father of taxonomy. He introduced binomial nomenclature. He made 2 kingdom classification. What are the 2 kingdoms? All of us know. One is plant kingdom, other one is animal kingdom. PLANATE and Animalia. He has done the classification. Now it did not distinguish. If you see plant kingdom, he kept bacteria also, protista also, fungi also under plant kingdom only on the basis of cell wall. He did not check the mode of nutrition. He did not see whether it is a prokaryotic cell or eukaryotic cell. He did not see whether it is unicellular or multicellular, photosynthetic or non-photosynthetic. He just saw cell wall is there, keep it under PLANATE. Cell wall is not there, bring it to Animalia. So these are the 2 kingdom classifications which was done by Carolus Linnaeus, who is a Swedish naturalist. Then comes 3 kingdom classification. I already wrote here. 2 kingdom classification did not distinguish between a primitive prokaryotic cell to an advanced eukaryotic cell. It did not distinguish between unicellular forms to multicellular forms. It did not distinguish between photosynthetic to non-photosynthetic forms. Then comes 3 kingdom classification, which is proposed by another scientist called Ernst Haeckel. Ernst Haeckel proposed 3 kingdom classification. Who is Ernst Haeckel? He is a German biologist. Ernst Haeckel proposed 3 kingdom classification. What did he do? He just separated all the unicellular organisms under a kingdom called Protista. Pro means first form. From unicellular, multicellular forms will come. So that's why what he has done, he has separated all the unicellular forms and he kept under one kingdom called Protista. So 3 kingdom classification includes 3 kingdoms. One is Protista. All unicellular forms are kept here, and PLANATE and Animalia. Still here he separated all unicellular organisms, but they are unicellular prokaryotic and unicellular eukaryotic, right? That distinction we cannot see in 3 kingdom classification. Then 4 kingdom classification came. Who proposed 4 kingdom classification? Copeland proposed 4 kingdom classification. What did he do? He separated the organisms which are under Protista into unicellular prokaryotic, unicellular eukaryotic. Unicellular prokaryotic are kept under the first kingdom. Mono means first kingdom called Monera. Bacteria which are unicellular prokaryotic are kept under the kingdom called Monera. Unicellular eukaryotic are kept in the kingdom called Protista. PLANATE, Animalia are there from the long back onwards. So that was the Copeland 4 kingdom classification. Unicellular prokaryotes under Monera, unicellular eukaryotes under Protista, then multicellular plants under plant kingdom, multicellular animals under animal kingdom. Then finally R.H. Whittaker. he's very popular. He proposed Phycomycetes classification in the year 1969. This point they have asked in NEET examination. R.H. Whittaker, in which year he has done the Phycomycetes classification? 1969. R.H. Whittaker has done Phycomycetes classification. Now what did he do? Now if you see fungi which are completely heterotrophic, since they are having cell wall, since they are multicellular, all these persons, Copland, Haeckel and Linnaeus, Aristotle, all these people, they kept fungi under plant kingdom only. How far it is true, tell me? Fungi are completely heterotrophic and plants are autotrophic, right? So they are completely or a diverse ends. So Whittaker, he finally separated fungi from the plant kingdom and he made it a separate kingdom and he proposed Phycomycetes classification. The first kingdom where unicellular bacteria are kept is called Monera. Unicellular eukaryotes are kept Protista. The third one which he separated is fungi which are heterotrophic. The next one is the plant kingdom and animal kingdom. He did Phycomycetes classification in the year 1969, right? Now he has taken 5 main criteria to classify the organisms under 5 kingdoms. The first one is cell structure, whether it's a prokaryotic cell structure or eukaryotic cell structure. Pro means primitive, karyon means nucleus. If they're having primitive type of nucleus, prokaryotic. If they're having true caryon nucleus, true nucleus if they have, eukaryotic. Now the second one is thallus organization, whether they are unicellular or multicellular. Monera includes unicellular prokaryotic organisms. Protista includes unicellular eukaryotic organisms. Fungi, Plantae, Animalia includes multicellular eukaryotic organisms. His first criteria is the cell structure, prokaryotic or eukaryotic. His second criteria is thallus organization, whether it is unicellular or multicellular. His third criteria is the mode of nutrition, whether it can prepare its food, autotrophic mode of nutrition, or it's depending on others, so heterotrophic mode of nutrition. His fourth criteria is the mode of reproduction, asexual mode of reproduction or sexual mode of reproduction. His fifth criteria is phylogenetic relationships. Ancestral relationships is called as phylogenetic relationships. On these 5 points, he has divided the living organisms into 5 kingdoms. Who is he? R.H. Whittaker. Take a screenshot, we will continue. So this is the table they have mentioned in NCERT textbook, table number 2.1, Whittaker's Phycomycetes classification. They tried to tabulate it. So I took it on the board. Let us see what are the 5 kingdoms what Whittaker has classified. The first kingdom is Monera, second kingdom is Protista, third kingdom fungi, fourth kingdom Plantae, last kingdom Animalia. He has done Phycomycetes classification on 5 characteristics. The first one is cell type. The second one is cell wall. Third one, presence or absence of nuclear membrane. Fourth one, body organization. And fifth one, the mode of nutrition. Now when we come to cell type, under these 5 kingdoms, there's only one kingdom which is having only prokaryotic organisms. Primitive type of cell, no nucleus. They are called prokaryotic type. Monerans comes under prokaryotic organisms, whereas Protista, fungi, Plantae, Animalia, they come under eukaryotic only. So they can ask us a question in Whittaker's Phycomycetes classification. How many prokaryotic kingdoms are there? The answer is 1, it is Monera. How many eukaryotic kingdoms are there? 4 kingdoms. Now the second point is cell wall. Now if we come to cell wall, Monera means where bacteria will be there, bacteria will have cell wall, right? So but it is not a plant cell like, it's not plant cell wall cellulosic. It is made up of polysaccharides, NAG and NAM, and it contains amino acids. So that's why they told cell wall is present, but it is not cellulose. It is made up of polysaccharides and amino acids. Now if we come to Protista, in Protista, Euglena will not have cell wall. Slime molds will not have cell wall. So that means there are some forms which have cell walls. So present in some forms of Protistans. Fungi, yeah, chitinous cell wall will be there. Then coming to plants, plants have cellulosic cell wall. Animal cell, no cell wall, only membrane will be there, right? Animal cell is delimited by the cell membrane itself. They don't have cell wall. Now the third criteria is nuclear membrane. Only prokaryotic cells, no nuclear membrane. Eukaryotic cells have well-defined nucleus and nuclear membrane. For the first kingdom, tell no. For all the remaining kingdoms, 2, 3, 4, 5, tell present, present, present and present. Now coming to the body organization. Now the first kingdom, bacteria, includes unicellular organisms. So cellular level of organization. Second kingdom, Protista, also includes unicellular eukaryotic organisms. But then organization, if it comes cellular only. When coming to fungi, it is multicellular. It's not unicellular. Fungi has hyphae, be, many cells will be there. So it is multicellular, loose tissue-like organization. Coming to plant kingdom and animal kingdom, plants have tissues. Tissues are organized into organs. Root, stem, leaf are organs. So it has multicellular, tissue and organ level of organization. Root, stem, leaves are organs. Coming to animals, the organs are further organized into organ systems, right? Digestive system, respiratory system, circulatory system, excretory system, reproductive system, like that. So organ system level of body organization is found in animal kingdom. Organ system is found in plant kingdom. Multicellular is found in fungi. Unicellular is found in first and second kingdom. Mode of nutrition, if you see, will tell bacteria are simple in structure, very small, but when it comes to nutrition, it has diversified mode of nutrition. It will do all types of autotrophic nutrition. It will do all types of heterotrophic nutrition also. Autotrophic means which can prepare the food. Heterotrophic means which depends on others. Under autotrophic, if it uses the sunlight, photoautotroph. If it uses the chemical energy, that is called chemoautotroph. Under heterotrophic, if it decomposes the dead and decaying organic matter, saprophyte. If it depends upon the living host, then it is parasitic, like salmonella. All these are parasites, right, on the intestine. E. coli is also a parasite. So mode of nutrition, bacteria shows autotrophic and heterotrophic. In autotrophic, if it is utilizing sunlight to prepare its food, photoautotroph. If it is using chemical, chemoautotroph, chemosynthetic autotroph. In heterotrophic, if it decomposes the dead and decomposing matter, saprophyte. And if it grows in the host, parasitic. Examples of bacteria are, examples of Monera are bacteria, primitive bacteria called Archaebacteria, and jokers of this thing we'll tell, they don't have cell wall, they are called Mycoplasmas. Mycoplasmas are also called PPLOs, pleuropneumonia-like organisms. So when we just summarize, Monerans, are they prokaryotic or eukaryotic? Monera is the only kingdom which is prokaryotic. Cell wall, it is non-cellulosic. It's made up of polysaccharides and amino acids. Nuclear membrane, it is absent. Only in Monera, nuclear membrane is absent. That's why it's called primitive karyotic. Body organization, it is unicellular. Mode of nutrition, diversified mode of nutrition. Examples are bacteria, Archaebacteria, Mycoplasma. Coming to Protista. Protista is unicellular eukaryotic kingdom. Some will have cell wall. Others, Diatoms and Dinoflagellates will have cell wall. Euglenoids and slime molds and protozoans will not have cell wall. Now body organization, it is unicellular only but eukaryotic. Mode of nutrition, when we tell, some are autotrophic, some are heterotrophic. Some are autotrophic members here, some are heterotrophic members here. Examples of protistants, just now I told, diatoms, dinoflagellates, slime molds, euglenoids and protozoans are the examples of the second kingdom, Protista. Coming to the third kingdom, fungi, they are eukaryotic. Now cell wall, yes, they have cell wall, chitinous cell wall. It's non-cellulosic cell wall. Coming to nuclear membrane, yes, it is also eukaryotic. True nuclear membrane will be there. Now body organization, multicellular, not unicellular, multicellular, hyphal network. Study of fungi is called mycology. Mode of nutrition, fungi is the only kingdom which are completely, exclusively heterotrophic. They cannot prepare their food. They are heterotrophic. And examples, if you see, Rhizopus comes under one class, then Penicillium comes under the second class, Agaricus comes under the third class, Trichoderma alternaria comes under the fourth class. So these are the examples of the fungal kingdom. Coming to the fourth kingdom, Plantae, they are eukaryotic. And if we talk about the cell wall, yes, cellulosic cell wall they have. Nuclear membrane, yes, they have. Level of organization, organ level of organization, they have. Root, stem, leaves are organs. Coming to the mode of nutrition, they are producers, means they are autotrophic. And examples, algae, bryophytes, pteridophytes, gymnosperms, angiosperms are the different groups of the plants. Coming to the animal kingdom, they are eukaryotic. They don't have cell wall. And nuclear membrane, they have. Level of organization, digestive system, respiratory system, circulatory system, means organ system level of organization. Mode of nutrition, heterotrophic, either holozoic or depending on the host, parasitic. Animal kingdom examples, chordates and non-chordates are the examples. Take a screenshot. We'll talk about the 5 kingdom classification merits and then we will get into the first kingdom, Monera. Alright, so this is there in NCERT textbook also, table number 2.1 it is there. Now let us talk about the merits of the 5 kingdom classification proposed by R.H. Whittaker. Now, in the remaining 2, 3, 4 kingdom classifications, now all Monera, Protista, fungi, all of them are kept in plant kingdom only, right? But in 5 kingdom classification, they separated unicellular prokaryotic under Monera, unicellular eukaryotic under Protista. Now, heterotrophic multicellular under fungi. Now, autotrophic multicellular under Plantae, and without cell wall they come under Animalia. So we can write down that earlier system included bacteria, cyanobacteria, fungi, ferns, gymnosperms, angiosperms under Plantae, since all of them have cell wall. So merits of 5 kingdom classification. What are the significant points of 5 kingdom classification? If we can tell earlier classifications include earlier systems included bacteria, cyanobacteria also, it included mosses, then it included ferns, it included bacteria, mosses, ferns, fungi, all of them under the kingdom called Plantae. With the fungi, it included gymnosperms also, angiosperms also under the kingdom called Plantae, because all of them are having cell wall. All of them has cell wall, so then they kept it under plant kingdom. But if you see the cell wall composition is made up of different materials, right? So this earlier classification, it has brought bacteria and cyanobacteria, which are unicellular, with other eukaryotic organisms, right? So prokaryotic should be separate and eukaryotic should be separate. So this earlier classifications have brought bacteria and cyanobacteria, prokaryotic. It has brought these prokaryotic organisms with other eukaryotic organisms, which is wrong. Whereas in Whittaker's classification, he separated it into different kingdoms. Now, earlier classifications also grouped unicellular organisms and multicellular organisms together. So, this is unicellular, these are multicellular. So, that's a drawback again. So, earlier classifications grouped unicellular organisms like Chlamydomonas, if you take, it's unicellular. It has included unicellular Chlamydomonas with multicellular forms. Earlier classification has done this, which is again wrong. In Whittaker classification, he separated unicellular organisms from multicellular organisms. Like in earlier classifications, Chlamydomonas and Spirogyra were kept under algae, right? And classification did not differentiate autotrophic plants and heterotrophic fungi. So till Whittaker's classification, fungi is in plant kingdom only. Earlier classifications did not distinguish multicellular heterotrophic organisms. It did not distinguish multicellular heterotrophic organisms from multicellular autotrophic organisms. Heterotrophic organisms fungi, multicellular autotrophic organisms means Plantae. So all these are demerits of the earlier classifications. When they have checked the cell wall, they didn't even check the composition of the cell wall. Plant cell wall is cellulosic, bacterial cell wall is polysaccharides and amino acids, fungal cell wall is chitin. They didn't even check the composition. Basing just they have the cell wall, bring it under Plantae. They don't have cell wall, keep it under Animalia. This was the earlier classification. Whereas in R.H. Whittaker's 5 kingdom classification, he brought all the unicellular prokaryotes under the first kingdom called Monera. He brought all the unicellular eukaryotes under the second second kingdom called Protista. He brought all the multicellular tissue level of organizing heterotrophic organism kingdom called fungi. He brought all autotrophic organ level of organization having cellulosic cell wall under the kingdom called PLANATE. And he brought the last kingdom, animals, which are, they don't have cell wall, they are eukaryotic, they are organ system level under the fifth kingdom called Animalia, right? So these are the 5 kingdoms what Whittaker has done. Now take a screenshot. We'll start with the first kingdom, which is Monera. Right, so let's start. So in this chapter we're going to look into the 5 kingdoms. The first kingdom is Monera. Now Monera means unicellular, right? They are unicellular and it's the only prokaryotic kingdom. It includes bacteria, cyanobacteria, mycoplasmas, actinomycetes. But even though there are so many types, bacteria are the sole members. Bacteria are the sole members. Ehrenberg coined the term bacteria. So when we talk about the first kingdom, which is Monera. Monera includes unicellular organisms. It includes prokaryotic organisms. Bacteria are the sole members, are the exclusive members of this kingdom. Bacteria are ubiquitous everywhere. They are seen everywhere. They are ubiquitous. They are present everywhere. So they are present in extreme harsh conditions also. They are seen in favorable conditions. Every organism will survive. But bacteria are even seen in extreme harsh conditions. Like for example, in hot springs also bacteria are seen. The bacteria which survive under hot conditions are called thermophiles. They are seen in cold conditions. On ice also they are found. Then they are called psychrophiles. Right? If they are found in saline conditions, salty conditions, they are called halophiles. Right? They are seen in hot springs, they are seen in snow, then they are called as psychrophiles. They are seen in saline conditions, halophiles. They are seen in deserts, dry conditions, xerophiles they are called, and many of them are parasitic. So if they are seen in desert conditions, dry conditions, they are called xerophiles. And if you see bacteria, most of them are parasitic. They cause diseases. Most of them are parasitic, means they cannot prepare the food, they depend on the host for the food. Then they are called parasitic. Now, who coined the term bacteria? Ehrenberg, E-H-R-E-N. Ehrenberg coined the term bacteria. Ehrenberg coined the term bacteria. Okay. Now, what are the 4 shapes of bacteria? If the bacteria is spherical shape, what is it called? If it is rod shaped, what is it called? If it is comma shaped, what is it called? If it is spiral shaped, what is it called? If the bacteria is spherical shaped, then we call it as cocci. If it is rod shaped, we call it as bacilli. Comma shaped, Vibrio. Spherical shaped, then they are called as spirillum. Now, cocci means this, right? Bacilli means rod shaped bacteria. Vibrio, comma shaped bacteria. Spirillum, this shape. If we want to take the examples of cocci, now monococcus, diplococcus, streptococcus, staphylococcus are the examples. Examples: monococcus, diplococcus, right? Streptococcus, staphylococcus. Examples of bacilli: lactobacilli, which converts milk to curd, is an example. Lactobacilli, which converts milk to curd. Now, comma shaped Vibrio cholerae, which causes cholera, is an example. Vibrio cholerae, which causes cholera, is an example. For spiral shaped bacteria, Treponema pallidum, which causes syphilis, is an example. For spiral shaped bacteria, what is the example I am giving? Treponema pallidum. What does it causes? Syphilis, which is sexually transmitted disease. Now, Vibrio cholerae, common shade bacteria, causes cholera. Vibrio cholerae is an example. Bacilli, lactobacilli, which converts milk to curd. And spherical shade bacteria, they are called as cocci. So, if one cocci is there, monococci. If two cocci are there, diplococci. If a chain of cocci is there, streptococci. If a bunch of cocci is there, like irregular grey bunch is there, this is called staphylococci. This is called streptococci. Now, if 4 cocci are there, then this is called tetrad. Now 4 in the front and 4 in the back, then this is called Sarcina arrangement. Then this is called Sarcina arrangement. Take a screenshot, then we will talk about the mode of nutrition. We told bacteria are very simple in structure, but they are very complex in the mode of nutrition because they are autotrophic also, they are heterotrophic also. Under autotrophic, we will tell they are photoautotrophs, they are chemoautotrophs. Under heterotrophic also, we will tell they are photoheterotrophs and they are chemoheterotrophs. Take a screenshot, we will talk about the mode of nutrition which is diversified in this kingdom. When we have to talk about the mode of nutrition of bacteria, we discussed that bacteria are most diverse in the mode of nutrition. The structure is very simple, but their mode of nutrition is very diverse. Let us see the mode of nutrition chart which I have taken on the board here. Now, depending on the energy source, whether they're taking energy from the sunlight or they're taking energy by oxidizing chemical compounds, right? So if they're taking the energy from the sunlight, call it as phototroph. If it is taking energy by breaking chemical compounds, then call it as chemotroph. Now, energy over. Now they have to take organic compounds also, carbon source also they need. Now depending on the carbon source, if the carbon source is organic compound, oxidation of organic compound, from there they get the organic compound, then they are called chemoheterotrophs. Chemotroph because energy source is chemical. Heterotroph because they are using organic compounds. They are oxidizing organic compounds to get the carbon. They are called chemoheterotrophs. Example for chemoheterotrophs are majority of the bacteria are chemoheterotrophs only. Most of the bacteria are chemoheterotrophs. Lactobacilli, which helps in conversion of milk to curd, is an example of chemoheterotroph. Antibiotic producing bacteria are also examples of chemoheterotroph. Nitrogen fixing bacteria, rhizobium, is also example of chemoheterotroph. And all this pathogenic bacteria like Salmonella, Shigella, Klebsiella, all these things, E. coli, they come under chemoheterotrophs only. Now coming to the other one, if the carbon source is atmospheric carbon dioxide, like plants they take atmospheric carbon dioxide. If the bacteria are taking carbon from atmosphere, they are chemotrophs because they take energy from the chemicals, but atmospheric carbon if they're taking, then they're called chemoautotrophs. So chemoautotrophs, the energy is derived from oxidation of inorganic compounds. Energy is derived from oxidation of inorganic compounds and carbon comes from atmospheric carbon dioxide. Then call it as chemoautotroph. Examples of chemoautotrophs are the bacteria which helps in conversion of nitrites, nitrates and ammonia, nitrosomonas, nitrobacter, nitrocystis, nitrococcus, all these are the bacteria. Chemoautotrophs play very important role in recycling of nutrients, in recycling of compounds like iron, phosphorus, nitrogen and sulfur. So we finished one set of classification. If we take the energy source as light, then that bacteria is called phototroph. Energy they get from sunlight. From where will they get the carbon again? Depending on the carbon source, if they take again the organic compound, if they break the organic compound for getting the carbon source, then it is called as photoheterotroph. Photoheterotrophs means energy from the sunlight, but carbon by oxidation of organic compound, then they are called photoheterotrophs. Examples for photoheterotrophs are Rhodospirillum and Rhodopseudomonas. Now, if they take carbon dioxide, atmospheric carbon dioxide, then call it as photoautotroph. Photoautotrophs means sunlight is an energy source and carbon dioxide is atmospheric carbon dioxide, then we call it as photoautotrophs. Now photoautotrophs, they do photosynthesis. When they are doing photosynthesis, are they releasing oxygen? Then call them as oxygenic photoautotrophs. If they are not releasing oxygen, anoxygenic photoautotrophs. Examples for oxygenic photoautotrophs are blue-green algae, chlorobium and chromatium. Examples for anoxygenic photoautotrophs are green sulfur bacteria, purple sulfur bacteria. So I think you understood the mode of nutrition reproduction in bacteria. So we are dividing them into two types depending on the energy source. If the energy source is chemical, it's a chemotroph. If they take the energy from the sunlight, then that is a phototroph. Now afterwards they need carbon also. If the carbon they are taking from oxidation of organic compounds, then it is called as chemoheterotroph. Organic compound, majority of the bacteria they are chemoheterotrophs only. Nitrogen fixing bacteria, pathogenic bacteria, curd making bacteria, antibiotic producing bacteria are the examples. Now energy source from the chemical, but carbon dioxide is atmospheric carbon dioxide, then called as a chemoautotroph. Majority of the recycling bacteria come under chemoautotrophs only. They get their energy from the chemical and carbon from atmospheric carbon dioxide. Example, Nitrosomonas, Nitrococcus, Nitrobacter. Now if they take sunlight as an energy, it's called phototroph. Now then carbon source, if they're getting from organic compounds, then it is called photoheterotrophs. Examples are Rhodospirillum, Rhodopseudomonas. If they get the carbon from atmospheric carbon dioxide, photoautotrophs. Photoautotrophs which release oxygen as a byproduct during photosynthesis, oxygenic photoautotrophs. Examples, blue-green algae, Chlorobium, Chromatium. If they do photosynthesis without releasing oxygen, then they are called anoxygenic photoautotrophs. Examples are green sulfur bacteria, purple sulfur bacteria. Take a screenshot, we will continue. Right? So now when we talk about bacteria, we told the word bacteria was coined by a scientist called Ehrenberg, and bacteria are unicellular, they are prokaryotic. Let us talk about the characteristics of bacteria. Now in the kingdom Monera, we have bacteria as the sole members. We have primitive bacteria which is called Archaebacteria. Then we have Mycoplasmas. Then next Cyanobacteria. So these are the examples of Monerans. Now in that we told the sole member is bacteria. Let's talk about few points about bacteria then. We have seen the shapes of the bacteria, 4 shapes of bacteria. Right? And then we have also discussed the mode of nutrition of bacteria. And they are unicellular and prokaryotic. Bacteria are unicellular, prokaryotic forms. Right? So bacteria, if they are locomotive, then they'll have flagella. If they are moving, then they move with the help of flagella. Bacteria move with the help of flagella. They move with the help of flagella. Now bacteria has cell wall. So bacterial cell wall is called murin. Bacterial cell wall, what is it called? It is called murin. Okay, it is REIN. It is called murin, which is made up of repeating units of NAG and NAM. Murin, which is also called peptidoglycan. It's also called peptidoglycan. The bacterial cell wall is made up of 2 sugars. What are the sugars called? NAG and NAM. The bacterial cell wall is made up of 2 complex sugars. One is called NAG, other one is called NAM. Repeating units, alternating units of NAG and NAM, linked by glycosidic bonds. NAG and NAM are linked. The sugars are linked by glycosidic bonds. NAG stands for N-acetyl glucosamine. NAG stands for N-acetyl glucose has amine, glucosamine. NAM stands for N-acetyl muramic acid. Now, it is a glycan part. Where is the peptide part? 4 amino acids will be attached to the NAM by peptide bond. So, first amino acid, second amino acid, third amino acid, fourth amino acid. Again, first amino acid, second amino acid, third amino acid, fourth amino acid. So, these are the amino acids. Since amino acids are there, peptide part is there. Since glucose, NAG and NAM are there, it's called peptidoglycan. This is a cell wall, composition of bacterial cell wall. Bacterial cell wall is called peptidoglycan, it is called murin, and it has cell membrane inside. The cell membrane of bacteria is made up of phospholipids. The cell membrane of bacteria is made up of phospholipids. Now, bacteria doesn't have cell membrane bound organelle, so no nucleus. Since it is not having nucleus, it is called prokaryotic. It's not having nucleus. If we draw a bacterial cell, in the bacterial cell, since nucleus is not there, the bacterial chromosome, it is circular and it is double stranded and it is naked. This is the genetic material of bacteria. It is having one chromosome which is circular and double stranded DNA. So since the DNA is naked, it is not wrapped by nuclear membrane, such a genetic material is called as nucleoid. It is also called ganophore. DNA of the bacteria, which is circular, double stranded, without the membrane, naked DNA, is called nucleoid or ganophore. Now, no nucleus and no membrane bound organelles. No membrane bound organelles means, what are membrane bound organelles? No mitochondria, no chloroplast, no Golgi, no ER, nothing. Any membrane bound structures will not be there in bacteria. Now, mitochondria is not there, then how bacteria will do respiration means, the plasma membrane enfolds to make structures which are called mesosomes. In bacteria, the plasma membrane is folded to make a mesosome. So what is this structure called? This is called mesosome. The mesosome is the enfolding of the plasma membrane which helps in respiration in the bacteria. So we have seen the cell wall, we have seen the cell membrane, we discussed with the mesosomes, then we talked about the genetic material, we told membrane bound organelles are absent. Now which type of ribosomes are present in the bacteria? The ribosomes of bacteria are of 70S type. Prokaryotic ribosomes are 70S, eukaryotic ribosomes are 80S, and they have reserve food materials in the form of glycogen. In bacteria, the reserve food material is glycogen bars. So what is this? This is a reserve food material which is called glycogen bar, and they contain inclusion bodies also. Bacteria contain, what are these? Inclusion bodies also. They contain inclusion bodies also. And bacteria divide by binary fission. Bacteria divide by binary fission. During binary fission, the bacteria splits into two daughter cells. The DNA gets replicated. One DNA comes this side and the other DNA goes this side, and eventually it divides into two daughter cells. This is asexual reproduction in bacteria, which is called as binary fission. We discussed everything related to bacteria, right? So we discussed like the four shapes, the mode of nutrition, photoautotrophs, chemoautotrophs, photoheteros, chemoheteros. We discussed that. Then we're telling bacteria, they are unicellular, they are prokaryotic, and they move with the help of flagella. Now the cell wall of bacteria is called murin, which is also called peptidoglycan. The composition is, it has repeating units of NAG and NAM. NAG stands for N-acetyl glucosamine, NAM stands for N-acetyl muramic acid. It has a tetrapeptide of 4 amino acids linked by peptide bond. Now the cell membrane is phospholipids. The cell membrane has infoldings which are called as mesosomes, which help in respiration. Any membrane bound structures are not there. No nucleus, no mitochondria, no Golgi, no ER is not there. Okay. Then next one, we tell that it has 70S ribosome. Bacterial ribosomes are of 70S type. Then they have reserve food material in the form of glycogen. They have inclusion bodies also. They show asexual reproduction by binary fission. Take a screenshot. Then we will talk about the next category which comes in Monera is Archaebacteria, primitive bacteria. Yeah, this is the information about bacteria. Now let's go to the next one. What is that? Archaebacteria. The meaning of archae itself tells like primitive. Archaeologist, archaeological survey we tell, means ancient, right? So Archaebacteria also comes under ancient bacteria, primitive bacteria. So ancient conditions are not good like this. They are harsh conditions, means Archaebacteria survived under extreme conditions. Harsh conditions means they should have certain peculiarities in the cell wall and cell membrane which made them to survive. So let's talk about Archaebacteria. What is the meaning of archae? Primitive. They are a category of primitive bacteria, ancient bacteria, which lived under extreme harsh conditions. Archaebacteria are primitive bacteria which lived under extreme harsh conditions. The ancient conditions are very harsh. Under those conditions it survived, so that's why we call them as Archaebacteria. I told you there should be some peculiarity in the cell wall and cell membrane which allows them to survive under that extreme harsh conditions. Now let us see Archaebacterial cell wall, what speciality it is. The cell wall of Archaebacteria is not murine, it is called pseudo murine. It is not murine, it is called pseudomurine. Pseudomurine means it doesn't have nag and nam, which is murine. It has nag and nat. Archaebacterial cell wall is made up of repeating units of nag and nat. Nag we know, N-acetyl glucosamine. What does NAT stands for? They are linked by glycosidic bonds. Again I need to write NAG, again I need to write NAT. Okay, so NAT stands for N-acetyl tallose aminouronic acid. So NAG we know, NAG stands for N-acetyl glucosamine, whereas NAT stands for N-acetyl tallose amuronic acid. NAT stands for N-acetyl tallose amuronic acid. Because of the presence of this component in the cell wall, the bacteria can survive in harsh and extreme conditions, we will tell. Right, understood? Now this is a difference in the cell wall. When we have to talk about the cell membrane, what difference we find in the cell membrane of the Archaebacteria? The cell membrane of Archaebacteria contains branched chain lipids, not plain lipids. They contain excuse me, they contain branched chain lipids, and they also contain carotenoids. The cell membrane contains branched chain lipids and carotenoids which will protect them from UV radiation's extreme harsh conditions. Now Archaebacteria are categorized into three groups. What are the three groups of Archaebacteria? One group of Archaebacteria which grows in the rumen of the cattle, they are called methanogens. The other group of Archaebacteria which grow in saline salt beds are called as halophiles. The third group of Archaebacteria which can survive at high temperatures, hot springs also, which can survive in acidic conditions, are also called as thermoacidophiles. The third category of Archaebacteria are called thermoacidophiles. So Archaebacteria are categorized into three groups. The first one is called methanogen, the second one is called halophile, the third one is thermoacidophile. Where are methanogens found? They are found in the rumen. Rumen is a part in the alimentary canal of the cattle. Methanogens are found in the rumen of cattle. Example of methanogen is Methanobacter, Methanococcus, Methanobacter, Methanococcus, all these are the methanogens are the examples here, right? Now, what do they do? Which type of organisms they are? They are strict anaerobes. Methanogens are strict means obligate. They are obligate anaerobes, means if you give little of oxygen also they will die. They are strict anaerobes. And then which mode of nutrition they show? They show chemoheterotrophic mode of nutrition. They show chemoheterotrophic mode of nutrition. Chemoheterotrophic mode of nutrition means carbon is by organic compounds and energy is by chemical. Both carbon compounds and energy, they take it from organic compounds oxidation only. So that's why they are called as chemoheteros. And this methanogenic bacteria only, they convert the organic acids into methane, biogas, gobar wax. Methanogenic bacteria help in biogas production, which is also called gobar gas production. So what did we study about methanogens? Methanogens, they come under Archaebacteria. They are seen in the rumen of the cattle. Now they are, examples are Methanobacter ruminatum, we can tell. They are strict anaerobes. They are chemoheterotrophic, means carbon also they get from organic compounds oxidation only. Energy also they get from oxidation of organic compounds only. They make biogas. Coming to halophiles, they grow in salt beds. Halophiles, where do they grow? They grow in salty beds. And these halophiles, which type? They are also obligate anaerobes only. Halophiles are also obligate anaerobes only, means strict anaerobes only. examples for halophiles which can survive under saline conditions, basic conditions. Examples are Halococcus, Halobacter. What are the examples? Halococcus. Next one, Halobacter. Now coming to thermoacidophiles, they survive at high temperature, they survive at acidic conditions, low pH. Thermoacidophiles are Archaebacteria which can survive at high temperatures. That's why they are called thermophiles. They can survive at acidic pH. Acidic pH means low pH. They can survive. So example is Thermoplasma. What's the example? Thermoplasma. Now, are they obligate anaerobes or facultative anaerobes? These two are obligate. Thermoacidophiles are facultative anaerobes, means they can survive in bit of oxygen. So they are not strict anaerobes. Thermoacidophiles are facultative anaerobes. Thermoacidophiles are facultative anaerobes. And which mode of nutrition do they show? These two show chemoheterotrophic mode of nutrition, but thermoacidophiles show chemoautotrophic mode of nutrition. Thermoacidophiles show chemoautotrophic mode of nutrition. What do you mean by chemoautotrophic mode of nutrition? Carbon from atmospheric carbon dioxide and energy by oxidation of inorganic compounds. Chemoautotrophs means carbon from atmospheric carbon dioxide, whereas energy from oxidation of inorganic substances. So this is about Archaebacteria. So what did we discuss about Archaebacteria? We told they are primitive bacteria. They survived under primitive conditions which are extreme harsh conditions. So what made them to survive means the cell wall and cell membrane. The cell wall of Archaebacteria is called pseudomurine, which is made up of repeating units of NAG and NAT. NAT stands for N-acetyl tallose amuronic acid. Now Archaebacteria cell membrane is made up of branched chain lipids and they contain carotenoids also. Archaebacteria are categorized into three bacteria types: methanogens, halophiles, thermoacidophiles. So these are the characteristics. With this, we finished bacteria, we finished Archaebacteria. Take a screenshot. We'll talk about Cyanobacteria then. Now, coming to Cyanobacteria. Cyanobacteria are also called blue-green algae, right? So examples of Cyanobacteria: nostoc, Anabina, spirulina we can take, then oscillatoria, right? So these are some examples. Trichodesmium, which causes Red Sea, is also Cyanobacteria. Now come on, let's discuss about Cyanobacteria, blue-green algae. So which do oxygenic photosynthesis like higher plants. They release oxygen into the atmosphere which we are taking and we are surviving. That's the respiratory gas for us. Now let's talk about blue-green algae, which is also called Cyanobacteria or blue-green algae. So we have unicellular forms also here, we have multicellular forms also here. Example for unicellular filamentous blue-green algae is spirulina. Example for multicellular filamentous is nostoc and Anabina. Let's write down examples of Cyanobacteria are, if we have to take the example of unicellular and filamentous, the example is spirulina. And whereas if we have to see the example of multicellular filamentous form, the examples are nostoc and Anabina. They help in nitrogen fixation, right? Now, which type of organisms they are? They are photosynthetic autotrophs. They are photoautotrophs. So, blue-green algae, they do photosynthesis. That means they are photoautotrophs. Photoautotrophs means, what do? They take atmospheric carbon dioxide, okay, and they take energy from the light, sunlight, and they evolve oxygen, right? So they release oxygen after photosynthesis, like higher plants. They release oxygen after photosynthesis. And what pigments do they have? They have chlorophyll A, which is similar to higher plants. They have chlorophyll A, which is similar to higher plants. That is why they are doing photosynthesis just like plants. They are doing photosynthesis just like plants. Now, you have seen the picture of nostoc they have given in the NCERT textbook. We can see the structure like it is covered by mucilaginous sheet. The colonies of the nostoc are surrounded by a gelatinous sheath. So, where are they found? They are found in aquatic conditions also, they are found in oceans also, they are found in terrestrial land conditions also. The body is covered by thin mucilaginous sheath or gelatinous sheath. We can tell the colonies of blue-green algae like nostoc and abina are covered by gelatinous sheath. They are covered by gelatinous sheath, and blue-green algae sometimes they grow profusely in the water bodies and they cause algal blooms. So blue-green algae can even cause algal blooms. They pollute the water bodies by causing algal blooms. Nostoc and Anabina, they help in nitrogen fixation. So in the structure of nostoc and Anabina, if you see in NCERT, they have been specialized cyst-like structures. What are they called? Echinids, heterocysts. So in these heterocysts and echinids, they help in nitrogen fixation. So Cyanobacteria, like nostoc and Anabina, they help in nitrogen fixation. The nitrogen fixing cells of nostoc and Anabina, what are they called? Heterocysts. So Cyanobacteria like Anabina and nostoc help in nitrogen fixation. They do nitrogen fixation under anaerobic conditions. So under aerobic or anaerobic, in absence of oxygen, under anaerobic conditions, they do nitrogen fixation. Now, those heterocysts will provide anaerobic conditions. Those structures are called heterocysts. In the heterocystic form, they do nitrogen fixation. Now, which type of reproduction they do? Sexual reproduction they go. Sexual reproduction, advanced organisms will go. So, we can tell sexual reproduction is absent. Asexual reproduction is by bi-interfission, we can tell. Sometimes fragmentation also happens. So, we can tell that in Cyanobacteria, sexual reproduction is absent. Sexual reproduction is absent. Now, if we have to talk about asexual reproduction is by binary fission. It is by binary fission. Sometimes fragmentation also occurs due to mechanical disturbance. Fragmentation also is seen in some cases, right? Then, if we have to see the differences between Cyanobacteria and bacteria, so I'll write it down. Differences between Cyanobacteria and bacteria. In Cyanobacteria, flagella are not there, but in bacteria, locomotor structures flagella are there. So write down no flagella, whereas here flagella are present, right? In Cyanobacteria, we can tell they do photosynthesis and they release oxygen, means the photosynthesis is oxygenic. Now in bacteria, it is anoxygenic photosynthesis. They do photosynthesis but they don't evolve oxygen. So Cyanobacteria, they do oxygenic photosynthesis, whereas bacteria will do anoxygenic photosynthesis. Bacteria will do anoxygenic photosynthesis. In bacteria, the reserve food material is glycogen. In Cyanobacteria, the reserve food material is starch, Cyanophycean starch. The reserve food material Cyanobacteria is Cyanophycean starch. The reserve food material of bacteria is glycogen. The reserve food material of bacteria is glycogen. This is one more difference. And bacteria, in bacteria, pigments are generally absent, whereas in Cyanobacteria, chlorophyll A is there, phycobilins are there, carotenoids are there. In bacteria, we can tell pigments are absent, whereas here we can tell chlorophyll A is similar to higher plants, carotenoids are also there, phycobilins are also there. So these are the differences between bacteria and Cyanobacteria. We finished bacteria, children. Then we came to Archaebacteria. We have seen the three types of Archaebacteria. The first two types are obligate anaerobes. The third one, thermoacidophiles, are facultative anaerobes. Then we came to blue-green algae, Cyanobacteria. Unicellular filamentous form example, Spirulina. Multicellular filamentous form example, Anabina and nostoc. They come under photoautotrophs, means just like plants, they take carbon dioxide from the atmosphere, sunlight as a source of energy. They release oxygen during photosynthesis. They do oxygenic photosynthesis. They have chlorophyll A, which is similar to higher plants. Now the colonies of blue-green algae, they are covered by gelatinous sheath. In NCERT, this diagram we can watch there. Now blue-green algae, sometimes they cause algal blooms and they pollute the water bodies. Cyanobacteria like nostoc and Animalia, they help in nitrogen fixation, but they'll do nitrogen fixation in absence of oxygen. So heterosis will create that anaerobic condition. Sexual reproduction is absent. Sexual reproduction is by biodiffusion or by fragmentation. Differences between cyanobacteria and bacteria. Bacteria has flagella, this will not have flagella. Bacteria will do anoxenic photosynthesis, cyanobacteria will do oxenic photosynthesis. It has cyanophysin starch as a reserve food material, bacteria will have glycogen as a reserve food material. In bacteria, the pigments are absent. Here chlorophyll A, phycobilins and carotenoids are present. Take a screenshot. We'll continue with blue-green algae again. Now when we have to talk about the examples of blue-green algae, we have seen nostoc and Animalia. We are seeing spirulina also. Oscillatoria also helps in nitrogen fixation in rice fields. Cyanobacteria helps nitrogen fixation in rice fields. Nostoc and Animalia can fix nitrogen in the free living state, in the soil also. They can fix nitrogen in the symbiotic form also. So nostoc is called as moon spit. These are the common names. It is called star jelly. Nostoc is called as moon spit. Nostoc is called as star jelly. It is also called fallen star. In that shape it will be there. So it is called as fallen star. And coming to spirulina, the dry weight of spirulina, 70% of the dry weight of spirulina is rich in protein. So we can consume spirulina as a source of protein. It's called single cell protein, right? And then we can tell they can change the light. They can change their color with what light they are receiving. That phenomenon is called guide-go phenomenon. So changing their color with respect to the light intensity is called as guide-go phenomenon. So cyanobacteria exhibits guide-go phenomenon. Not only that, Trichodesmium erythraeum will grow and make the sea appears red. So the sea is called Red Sea. What is that cyanobacteria which makes Red Sea? Means it is Trichodesmium erythraeum. It causes a Red Sea. It grows and it makes the sea appear red. It is called as Red Sea. All this is information about cyanobacteria. So we finished bacteria, we finished Archaebacteria, we finished cyanobacteria. Take a screenshot. We will continue. In Monera, the last group of organisms are Mycoplasmas. So what is the speciality of Mycoplasmas? Means they are smallest in the kingdom of Mycoplasmas. Another speciality of Mycoplasma is they don't have cell wall. Since they don't have cell wall, they can change their shape often. Means they are pleomorphic. That's why Mycoplasmas are also called as jokers of plant kingdom. When it comes to discovery, who discovered Mycoplasmas? Means Nocard and Rox are the names of the scientists who discovered Mycoplasmas. They are very simple. They are small, they are free living, they are unicellular, and they are anaerobic organisms without cell wall. Let us write down the characteristics of Mycoplasmas. So Mycoplasmas are smallest prokaryotes. They are smallest prokaryotes. They are unicellular. They are simple in structure. They are free living. And they are anaerobic also, right? They don't have cell wall, no cell wall. Since cell wall is not there, they can change their shape. Means since cell wall is not there, they keep changing their shape. That means they are pleomorphic. So that's why since they are changing their shape, we can also call them as jokers of plant kingdom. spleo, right? Since they keep changing their shape, we can also call them as jokers of plant kingdom. If you remember, Monera was once kept under plant kingdom only. Then they named Mycoplasma as jokers of plant kingdom. And they were discovered by two scientists, Nocard and Rox. Who discovered it? Nocard and Rox. Discovered Mycoplasmas. They discovered Mycoplasmas. What are the other names of Mycoplasmas? They are also called MLOs. They are also called PPLOs. They are also called MLO. They are also called PPLOs. What do you mean by MLO? Mycoplasma-like organisms. PPLO, pleuropneumonia-like organisms. The other name of Mycoplasmas are Mycoplasma-like organisms. And the other name is, they cause pneumonia, pleuropneumonia. That's why they're called pleuropneumonia-like organisms. Pleuropneumonia-like organisms. Since they don't have cell wall, like there's no action of lysozyme, there's no action of penicillin or Mycoplasma, because lysozyme breaks the cell wall. Penicillin also stops the synthesis of new cell wall. Since Mycoplasmas don't have cell wall, there's no action of lysozyme and penicillin on Mycoplasmas. There is no action of, one is penicillin and lysozyme enzyme on what? On these MLOs, Mycoplasma-like organisms. Since they don't have cell wall, these two things, penicillin and lysozyme, cannot act on Mycoplasmas. And what do they cause? They cause some diseases. They are pathogenic. They cause diseases in plants also. They cause diseases in animals also. They are pathogenic. What diseases they cause in plants? What diseases Mycoplasma cause in animals? The disease what Mycoplasma cause in plants is called as witch's broom in wooden plant. It is called witch's broom, where in woody plants or in woody trees we can tell. So this is a plant disease. Coming to animal disease, we can tell pleuropneumonia is one disease and urethritis, urinary infection, is the other disease. So animal diseases, one disease is pleuropneumonia and the other disease is urethritis. Okay, now Mycoplasmas, they can be considered as gram-negative bacteria, gram-negative organisms, and they are non-motile. If we have to list down one more character, we can tell Mycoplasmas are, they belong to gram-negative type and they don't have any motile structures, means they are non-motile. They are non-motile. So with this, we have seen different organisms which come under bacteria or Monera. We have seen bacteria, we have seen Archaebacteria, we have seen Cyanobacteria, and then we have seen Mycoplasmas also. Mycoplasmas, they are the smallest prokaryotes. They are very small. They are unicellular. That's why they are kept under prokaryotic kingdom. They are simple, they are free living, and they are anaerobic. Since they don't have cell wall, no cell wall, since they don't have cell wall, they keep changing their shape. They're called jokers of plant kingdom. And the other names are MLOs and PPLOs. Since they don't have cell wall, there will not be any action of penicillin or lysozyme on Mycoplasmas. The pathogenic forms means what diseases they cause in plants means witch's broom in woody trees. In animals, it causes pleuropneumonia and urethritis. Take a screenshot. We'll talk about the how microorganisms or bacteria are useful in beneficial activities and how they cause pathogenic activities. Now, let us conclude this kingdom Monera by talking about the beneficial activities or harmful activities of bacteria. Bacteria are the sole members of Monera, no? Now, bacteria, we call their friends also, their foes also. Means they have some beneficial activities also, they have some harmful activities also. Harmful activities means they are pathogenic. They cause diseases like cholera is caused by bacteria only. Typhoid is caused by bacteria only, right? So, how do they reproduce? Asexual reproduction in bacteria is by binary fission. So, reproduction in bacteria, if we talk, reproduction in bacteria, if you see, one method is asexual reproduction right? Asexual reproduction is by binary fission. It is by binary fission. In binary fission, what happens? The bacterial cell splits into two daughter cells. One DNA copy comes here, other DNA copy goes here, and eventually they divide into two daughter cells equal in size. So this is binary fission. So one method of asexual reproduction is by binary fission, and sometimes during unfavorable conditions the bacteria can produce spores also. So during unfavorable conditions, so bacteria make spores. They produce spores to tide over unfavorable conditions. Coming to sexual reproduction, bacteria exhibit primitive type of sexual reproduction which comes under gene transfer mechanisms like transformation, transduction and conjugation. This is a sexual reproduction. When we have to talk about sexual reproduction, so we need to tell that bacteria exhibits primitive type of gene transfer mechanisms. Primitive type of gene transfer mechanisms. How many types of sexual reproduction do they show? Means 3 types. So first method is called transformation. The second method is called transduction. The third method is called conjugation. The 3 types of sexual reproduction are transformation, transduction and conjugation. Now transformation means it is uptake of DNA by the bacteria. In the surrounding some DNA is there, and if the bacteria takes the DNA from the external medium, this is external medium. From the external medium, if the bacteria takes the DNA, that technique is called transformation. What is transduction? Now, if the DNA is coming through a virus, this is virus. The virus has infected a bacterial cell. This is virus. The virus has infected a bacterial cell and virus is sending its genetic material inside. If the virus sends the genetic material inside, that's called transduction. Transfer of genes through a virus is called transduction. Transfer of genes from outside is called transformation. Conjugation. During conjugation, 2 bacteria, they will come close to each other and these 2 bacteria will have sex pili. Through their sex pili, they make conjugation tube. Through the conjugation tube, they exchange the DNA. That is conjugation. So these are primitive types of sexual reproduction, because we are not talking about male and female. We are not talking about gametes and all. It's a primitive type of gene transfer. Here the gene is transferred from the external medium. Here the gene is transferred via virus. Here the gene is transferred via conjugation tube. These 3 are different types of sexual reproduction. Coming to why we call bacteria as friends and foes, means we need to talk about the beneficial activities, we need to talk about the harmful activities. So under beneficial activities, we can tell Rhizobium bacteria helps in nitrogen fixation, Lactobacilli helps in curd production, right? Curd is a probiotic, and we can also tell, like, from bacteria we can extract many antibiotics. We can extract many antibiotics. So 1, 2, 3, so many other, we just listed some beneficial activities of bacteria. Coming to foes, means what are the harmful effects of bacteria? Means they cause diseases, they are pathogenic. For example, cholera is caused by Vibrio cholerae, typhoid is caused by Salmonella typhi, tetanus is caused by Clostridium tetani. These are animal diseases. Under plant disease, citrus canker is caused by Xanthomonas citri. I took those diseases which are there in NCERT, right? So under foes, means we have to tell, the foes means pathogenic. Under pathogenic, if you have to list down the animal diseases, cholera is one animal disease, typhoid is another animal disease, tetanus is another animal disease. Cholera is caused by Vibrio cholerae. Typhoid is caused by Salmonella typhi. Tetanus is caused by Clostridium tetani, right? So these are animal diseases. Under plant diseases, we can tell citrus canker. Citrus canker is the disease caused by Xanthomonas citri, by a bacteria called Xanthomonas So we tried to cover every information about the kingdom Monera. Now let us start with the second kingdom called Protista. Take a screenshot. Now we finished the first kingdom under Whittaker's 5-kingdom classification, that's Monera. Now let's go to the second kingdom. The second kingdom is Protista. Now proto, what's the Greek meaning of protos or proteos? Means first of all, right? So unicellular forms evolved first, later multicellular forms came from unicellular. So proteos means first formed unicellular organisms, and the kingdom Protista was coined by Ernst Haeckel, who did 3 kingdom classification, right? So let's go to the kingdom called Protista. Now the Greek meaning of Protistos means first of all. The Greek meaning of Protistas means first of all. These are the first formed organisms, and the word Protista, the kingdom Protista, was introduced by Ernst Haeckel. So the kingdom Protista was introduced by Ernst Haeckel. It was introduced by Ernst Haeckel. Now if you see, like here we have the first kingdom Monera, which is unicellular prokaryote, and the other side if you see, we have multicellular eukaryotes. We have multicellular eukaryotic kingdoms like fungi, PLANATE, Animalia. Now one end we have unicellular prokaryote, the other end we have multicellular eukaryotes. In between there is a bridge which is called as Protista. Now Protista is unicellular, means in unicellular nature it resembles bacteria, and it is eukaryotic. Eukaryotic nature matches these kingdoms. So can we call Protista acts as a connecting link between unicellular forms to multicellular forms. Protista acts as a connecting link between prokaryotic forms to eukaryotic forms, right? So let us write down. So we can tell that Protistants act as connecting links between unicellular forms to multicellular forms, or we can tell they act as connecting links between prokaryotic forms to eukaryotic forms. Since they have these characters also, the boundaries for this kingdom cannot be clearly defined. The boundaries for this kingdom cannot be clearly defined because they possess these characters also, they possess those characters also. So let us write down boundaries of this kingdom are not clearly defined. We cannot clearly define the boundaries because they have these characters also and these characters also. Now for a botanist it might appear like a plant, but for a normal scientist it appears like a protestant. So what may be a photosynthetic protestant might appear a plant for a botanist. So we can tell what might be a photosynthetic protestant might appear, it might appear plant. To whom? Botanist. Now you know, if you take the form Euglena, now Euglena, it will do photosynthesis. For a botanist, Euglena he'll keep under plant kingdom, but for us it is protestant, right? Euglena is protestant, Paramecium is protestant. Paramecium, they can take it as animal kingdom since it is moving. Euglena, since doing photosynthesis, botanists tell it is plant, but actually Paramecium doesn't come under animal kingdom and Euglena doesn't come under plant kingdom. Both of them, they come under Protista, right? So then if we see, are they aquatic or they terrestrial, means most of them, primarily they are aquatic, means mostly they are aquatic. We can tell primarily they are aquatic forms. They are aquatic forms and they are unicellular, multicellular, they are unicellular, and since they are unicellular, since they are eukaryotic, let us write down, they are unicellular forms. They are eukaryotic form. Since they are eukaryotic, they will have all membrane bound organelles. Since they are eukaryotic, they'll be having all membrane bound organelles, like they have well defined nucleus, they have mitochondria, they have Golgi, they have ER. So all the eukaryotic characteristics they have, even though they are unicellular, they have all the eukaryotic characteristics of well-defined nucleus. So they have mitochondria, Golgi, ER, all these things they have, and they have cytoskeletal structures also, and they show cytoplasmic streaming also. Cytoplasmic streaming means movement within the cell. So they have cyclosis, they exhibit cyclosis. Cyclosis means if the contents of the cell move within the cell, this is called cyclosis. They exhibit cyclosis. This type of movement also they show, and they have cilia and flagella also. If you have to see for Euglena, Euglena has one big flagella and one small flagella. And we have to see for Paramecium, Paramecium has cilia all throughout the body. So they exhibit cyclosis movement also, and they have locomotor structures like flagella, and they have locomotor structures like cilia. And I told you, like Euglena has, it will do photosynthesis since it has ribbon like chloroplast. So these are the chloroplast which I am drawing. So they have chloroplast, they have chloroplast, and they are photosynthetic. So we came to the second kingdom, Protista. Let's just summarize. The meaning of proteose means, first of all, these are the first unicellular organisms. The term Protista was introduced by Ernst Haeckel. Now this Protistans, which comes under second kingdom, unicellular nature matches with Monera. Then next one, eukaryotic nature matches with fungi, PLANATE and Animalia. Since it is possessing this kingdom characters and the other kingdom characters also, we tell they act as connecting links, and they don't have any boundaries. Boundaries cannot be clearly defined. So for us, this is a Protistan, but for a botanist, since it's doing photosynthesis, they'll call it as a plant. For us, this is a Protistan. Paramecium is a Protistan, but since it is having cilia and it's locomotory, like a zoologist may call it as an animal. Coming to the characteristics, most of them are aquatic, they are unicellular, they are eukaryotic, they possess all the membrane bound organelles, they exhibit cyclosis movements also, they have flagella as a locomotor structure, they have cilia as a locomotor structure, they have chloroplast, and they are photosynthetic. Take a screenshot, we will continue. Now when we have to talk about asexual reproduction and mode of reproduction in Protistans, they do asexual reproduction also and they do sexual reproduction also. Asexual reproduction is by fragmentation, and sexual reproduction is by cell fusion and zygote formation. And you know how many groups are there in the Protista? There are 5 groups in Protista, which they have done in NCERT textbook. So let us write down which type of asexual reproduction does it exhibits. Which type of sexual reproduction does it exhibits? I told asexual reproduction is by binary fission, and sexual reproduction is by cell fusion and zygote formation. These are the sentences which are there in NCERT. Asexual reproduction in Protista is by which method? If they ask, the answer is binary fission. Sexual reproduction involves cell fusion, gamete fusion. If the gametes are fused, zygote will be formed. So cell fusion and zygote formation. Now how many groups are there in Protista? There are 5 groups in Protista. The first group is called as Chrysophytes. The second group is called as Dinoflagellates. The third group is called as Euglenoids. The fourth group is called as slime molds, right? And the fifth group is called as Protozoan Protists. So how many groups are there, children? There are 5 groups under Protista. The first group is called Chrysophyta. The second group is called Dinoflagellata. The third one, Euglenophyta or Euglenoids. The fourth group is slime molds. And the last one is Protozoan Protists. Now let us talk about Chrysophytes. Chrysophytes, chryso means golden color. Chrysophytes include diatoms and desmids. So again under Chrysophytes, we are telling it includes diatoms and desmids. It includes diatoms and desmids. Desmids are called as golden algae. Desmids are called golden algae. And chrysophytes, they live in freshwater also, they live in marine water also. So habitat in freshwater also, as well as we can tell they'll be found in marine water also. Why is it called as diatom? Means, so the body of these chrysophytes or diatoms will be like a soap box. It has an upper shell and it has a lower shell. Diatoms, the body is covered by two halves. This is the upper shell and this is a lower shell. So this is a upper shell and this is a lower shell. And isn't it appearing like a soap box? That's why we call it as diatoms. There are no locomotor structures in diatoms. No, I did not draw any locomotor structures here. No cilia, no flagella, no locomotory structures in diatoms. Diatoms are planktonic, means they can do photosynthesis, they float on the upper surface. So they are planktonic. They are planktonic, they are photosynthetic. So they will grow nicely in the water. So that's why we call them as pastures of the sea. Chrysophytes, diatoms are called as pastures of the sea. In the sea water, they spread nicely, they do photosynthesis, they are planktonic, they don't have any locomotor structures. The body is covered by shell, upper shell and lower shell. Inside it has a large central vacuole. Inside, if you see, it has a large central vacuole. Inside the large central vacuole, suspended nucleus will be there. Since it is eukaryote, it has its nucleus. It has a large central vacuole. Let me write down. It has a large central vacuole, and in vacuole or inside vacuole I am drawing nucleus. Inside vacuole, nucleus is suspended by cytoplasmic strands. The cytoplasmic strands, you know, they help in placing the nucleus perfectly here. It has all the eukaryotic organelles as usual, right? And then we can tell like these diatoms and desmids, or we can tell the diatoms act as the chief producers in the ocean. They are the chief producers in ocean. We told they are photosynthetic, they are planktonic. So diatoms are chief producers in the ocean, and the diatom cell wall contains silica. So this cell wall, right, the diatom cell wall contains silica, and this silica is indestructible. It will not destroy. Since the silica is indestructible, when the diatoms die, they go and settle at the bottom. They go and settle at the bottom, and this silica will make a silica ores. This silica ores, which is mixed in the sand, this sand is used for filtering oils. It is used as a glossy paint. We call it as a diatomaceous earth. When the diatoms die, the cell wall which is having silica, this silica is indestructible. It goes to the bottom and it's called diatomaceous earth. This diatomaceous earth or diatomite, where is it used? It is used in filtration of oils, filtration of syrups. It enhances the night visibility also. It is used in polishing also. It is used in filtering oils. It is used in filtering syrups. It is used in glossy paints, and it helps in polishing. And it helps to prepare night visuality. In night, this diatomaceous earth, all that glass and all will shine. Night visibility, it will increase. Night visibility will increase. So these are the points about chrysophytes. Understood, children? We told protistans, they have 5 groups. The first group is called as chrysophyta. Chrysophyta includes diatoms and desmids. Why are they called as diatoms? Because the body is having 2 parts. The other group is called desmids, which are called as golden algae. They are in fresh water also, they are in marine water also. The body appears like a soap box. They don't have any locomotor structures. They have a large central vacuole. Inside the vacuole, nucleus will be suspended. They will have all the organelles, and they are photosynthetic, they are planktonic, they are called pastures of the sea, and they are the chief producers in the ocean. And since the cell wall is having silica, the silica is indestructible and it makes diatomaceous earth. This diatomaceous earth is used in making polishes, is used as a filtration agent, it is used as an insulator, it is used for night visibility. All this is information about the first group. Take a screenshot, then we will talk about dinoflagellates. Now what's the second group? Dinoflagellates, right? So the name itself tells dino means two, flagella. Diatoms don't have flagella, but dinoflagellates have flagella. Now the body of dinoflagellate will be like this. It has a longitudinal furrow and it has a transverse girdle, and the body, it has ornamental designs on it. It has ornamental designs on it. Now it has two flagella. Dinoflagellate, right? It has two flagella. One flagella is the longitudinal flagella. The other flagella which goes in the girdle, this is the transverse flagella. There are two flagellas, and these two flagellas will rotate like a top. So that's why they're called phyalgae. They're called as phyalgae. They're also called as spinning flagellates. They're also called as spinning flagellates. Right? So this is about dinoflagellates, and dinoflagellates, they're mostly marine. They're seen in marine forms only, marine water bodies only. There are some dinoflagellates like Gymnodium and Gonilex. They grow profusively in the Mediterranean Sea and make the sea appear red, and this is called as red tide. So there are two examples of dinoflagellates. What is that? Gymnodium and Gonilex. Gymnodium is one. Gonilex. Gymnodium and Gonilex, they grow in Mediterranean Sea. They grow in Mediterranean Sea and they cause red tides. Okay, so this point is very important. Many times they have asked this point in NEET examinations. So dinoflagellates, they are mostly marine, and the body has frustules. It has frustule-like body. It has two grooves. One is a longitudinal groove. Longitudinal flagella will be there. One is a transverse groove in which transverse flagella will be there, and they rotate like a top. That's why they're called as phyalgae. They're also called spinning flagellates, and we have to talk about they are also photosynthetic. They are also photosynthetic. Their chief pigment is chlorophyll A, since it is eukaryotic. So they have all membrane-bound organelles. They have all membrane-bound organelles. The main pigment is photosynthetic A. Apart from photosynthetic A, apart from chlorophyll A, they have chlorophyll B also. They have xanthophyll also, right? And carotenoids also. Because of the xanthophyll and carotenoids, now dinoflagellates will be in various colors. They'll be in blue color, they'll be in green color, they'll be yellow, they'll be orange, they'll be brown, or these are the different colors of dinoflagellates. Take a screenshot, we will continue. Under Protista, we finished Chrysophytes, we finished dinoflagellates. Now the third group is Euglenoids. Let us discuss about the third group under Protista. They are called Euglenoids. Under this, we can take Euglena as an example. What is the speciality of the Euglena? Euglena acts as a connecting link between plant kingdom and animal kingdom. It acts as a plant also, it acts as an animal also. So what is Euglena? So Euglena is connecting link. It is connecting link between plant and animal, plant kingdom and animal kingdom. Why are we telling that? It's a connecting link between plant kingdom and animal kingdom. If you see the body of Euglena, it will be in this manner. This is the body of Euglena, and this one is the mouth, which is called cytostome. The mouth leads to a region, this portion is called cytopharynx. Cytopharynx leads to next compartment, which is called reservoir. And at the reservoir, this side, if you see, it has an eye spot, which is called stigma or eye spot. Eye spot it is having, so it is animal, right? It has eye spot, and then from here it has two basal bodies. From the basal body, from one basal body, small flagella, from the other basal body, long flagella will arise. So what is this? This is basal body. From the basal body, flagella, one small and one long flagella will arise, right? and then we here it has a contractile vacuole. This is a contractile vacuole. All the aquatic forms, in order to regulate osmosis, they have contractile vacuoles. So this we can write it as contractile vacuole. Another specialty of Euglena is the body of Euglena is not covered by cell wall, it is covered by a flexible protein pellicle. So this is, what is this? This is flexible protein pellicle, right? And this pellicle is arranged in this manner. Obliquely the pellicle will be there all throughout the body. Since it is having eye spot, we're telling it's an animal character, and it has ribbon-like chloroplast inside. So ribbon-like chloroplast. So these are chloroplast. If it is having chloroplast, it can do photosynthesis, means this is a plant character. Having eye spot is an animal character. It is an animal character. Having mouth, when sunlight is there, it will do photosynthesis. So then it's a plant character. When sunlight is not there, so it engulfs. It will do holozoic mode of nutrition. It engulfs pathogens, holozoic mode of nutrition. Animal mode of nutrition. When it will do holozoic mode of nutrition? In absence of light. In presence of light, it is doing photosynthesis. In absence of light, it is eating the food materials. That means it is having both plant character and animal character, right? And in presence of light, it will do photosynthesis. It will prepare starch. The starch is stored here as paramyelin starch granules. So here itself, it will store the starch granules. What are they called? Paramyelin starch granules. Okay, and it has a big nucleus at the center. So this is the nucleus. Since it is eukaryotic, it has nucleus with prominent 1 or 2 nucleoli. So it has 1 or 2 nucleoli, and this is a chromatin. This is chromatin. So these are characteristics of euglenoids. And where are euglenoids found? Euglenoids are found in freshwater, that too stagnant freshwater, not flowing water bodies. So euglenoids are found in freshwater, and they are found in stagnant water bodies, not floating water bodies. They are found in stagnant water bodies. And the division of Euglena, asexual reproduction, they go, it's a longitudinal fission. So asexual reproduction, it shows asexual reproduction is by longitudinal binary fission. At the time of binary fission, it loses the flagella and then it divides into 2. So it loses flagella and then the body is dividing into 2, and it splits like this. Then it will become 2 daughter euglenas. This is one daughter euglena and this is another daughter euglena. When you split this, it becomes 2 daughter euglenas. Later it will generate its flagellas, and this is called longitudinal binary fission. And they have chlorophyll, they have chloroplast, they have chlorophyll, it has plant characters, it has animal characters. So this is about the third group under Protista, which is called as euglenoids. Take a screenshot. We'll talk about the next group, which is called as slime molds. Now the fourth group under Protista is slime molds. Now the slime molds means they are mold-like, means they have saprophytic mode of nutrition. Let's write down the fourth group. What is it? Slime molds. So this group, earlier euglenoids, they are having photosynthetic mode of nutrition. These will have saprophytic mode of nutrition. Saprophytic mode of nutrition. And these slime molds, in favorable conditions, they also don't have cell wall. In favorable conditions, they spread over the decaying twigs and all. Several feet they spread, and they form aggregates, masses, which is called as plasmodium. When do they form aggregates? When do they spread over several feet? Means under favorable conditions. So we can tell during favorable conditions, during favorable conditions, the slime molds, they spread several feet. They are spreading several feet. Where are they spreading? On the decaying twigs. They are spreading several feet on the decaying twigs and they make aggregates. What are the aggregates called? Plasmodium. During unfavorable conditions, what it will do? During unfavorable conditions, now this plasmodium undergoes asexual reproduction. They form fruiting bodies. On the fruiting bodies, spores will be there. These spores will be having true walls. Because of the presence of true walls on the spores, they can withstand any unfavorable condition for years together. They can sustain. So under favorable conditions, it's forming aggregate, which is called plasmodium. It spreads several feet on the organic matter and it engulfs the organic matter. Whereas during unfavorable conditions, if we have to talk, during unfavorable conditions, what will happen? This plasmodium breaks. This plasmodium changes into a fruiting body, and this fruiting bodies on the top, they have spores. Now these spores have thick true walls. Because of the presence of these thick true walls, the spores can tolerate, they can tolerate adverse conditions for years together. Nothing will happen. Till the favorable conditions come, they can retain, they can sustain within the spores, and when the favorable conditions comes, the spore wall breaks and again the body, the slime mold body comes out. So this is information about the fourth group of Protista, which is called as slime molds. What are slime molds? They are saprophytic protists. During favorable conditions, they make aggregates. What is that called? Plasmodium. During unfavorable conditions, it undergoes asexual reproduction and it produces fruiting body. On the fruiting body, they produce spores. The spores will have thick walls. These thick walls will allow the spores to sustain for years together adverse and dormant conditions. Take a screenshot. We'll talk about the fifth group under Protista, which is called as Protozoan Protists. Now earlier these Protozoan Protists were kept under animal kingdom because of the animal mode of nutrition. Since animal kingdom should be having only multicellular forms, these Protozoan Protists are unicellular forms. They brought it back to Protista. Haeckel brought it back to Protista. Now the last group under Protista is Protozoan Protists. Under Protozoan Protists, we have four groups again. What is the first group, second group, third group and fourth group? The first group, they are called as amoeboid protozoans. The second one are called as flagellated protozoans. The third one are called as ciliated protozoans, and the last one are called as sporozoans. Basing on their locomotor structure, we have divided the Protozoan Protists into four types. If they have amoeba-like body, if they have pseudopodia, then we should call them as amoeboid protozoans. If they're having flagella, call them as flagellated protozoans. If they're having cilia as a locomotor structure, ciliated protozoans. If they're not having any locomotor structure, then they're called as sporozoans. So basically, these Protozoan Protists are microscopic, they are unicellular, most of them are parasitic. Why? They are non-photosynthetic. They are non-photosynthetic, they are parasitic, some have locomotor structures, some doesn't have locomotor structures. The first group I told, it is amoeboid protozoan. In amoeboid protozoan, we can take the example as Entamoeba histolytica. Entamoeba histolytica comes under amoeboid protozoan, which is unicellular, and it causes amoebic dysentery, right? It causes amoebic dysentery in humans. This Entamoeba histolytica, so the body will have many pseudopodia. This is one pseudopodia, another pseudopodia. Since it is prokaryotic, it's eukaryotic, it has a well-defined nucleus and it has cytoplasm. So this is an example of amoeboid protozoan. So these are the pseudopodia, they arise, and their cell walls, they have silica shells. The marine amoeboid protozoans, what do they have? They have silica shells. diacetylica cells. Coming to the second one, flagellated protozoan. Under flagellated protozoan, we can take the example of trypanosoma. What's the example? Trypanosoma gambiense, which causes African sleeping sickness. Trypanosoma gambiense causes African sleeping sickness. And if we see the body of trypanosoma, it will be like this. So it has flagella here. The flagella from this end to that end it goes, and the flagella fuses with the body. So this flagella, if you see, it is arising from the basal body and it is fusing with the body wall here, and it forms a membrane, and this is a free end of the flagella. It has nucleus, all other structures it has. This is Trypanosoma gambiense. The body will be flexible, it doesn't have any wall, it is unicellular, it is parasitic, it causes African sleeping sickness. It has one flagella which arises from anterior side to posterior side. Anterior, the flagella is fused with the body wall to form a membrane. This is called undulating membrane. And this is a free end of the flagella. Posterior end is a free end. What does it cause? It causes African sleeping sickness. Coming to the third class, ciliated protozoans. Under ciliated protozoans, we can take the example of paramecium. Paramecium is looking like a slipper, so we call it a slippery animalcule. It is like a slipper, it's a small animal, so we call it a slippery animalcule. If you see the body of paramecium, exactly it appears like a slipper. Right, so here is a mouth, it is called cytostome. The cytostome leads to this structure which is called gullet. The body all throughout it is covered by cilia, whereas the cilia at this end will be very long. At the caudal end, the cilia are very long. The cilia move together in a rows. They help in locomotion of the food also. They help in locomotion also. They help in gathering the food also. These cilia are called caudal tuft. Posterior end cilia are very long. They are called caudal tuft. All the cilia will move together like oars. And this is cytostome. Cytostome leads to gullet here, and it has anterior contractile vacuole. It has posterior contractile vacuole also. This is the anterior contractile vacuole, and here is a posterior contractile vacuole. And it has dimorphic nucleus, one macronucleus and one micronucleus. Here is the macronucleus it has. This is a macronucleus and this is a micronucleus. Having two nuclei, different types of nuclei is called as dimorphic nuclei. Children, this question they asked in NEET 2018 examination. They asked, dimorphic nucleus is present in which class of Protista? It is present in Protozoan Protista under ciliated protistans. Under ciliated protistans, example is paramecium. Paramecium is called slippery animalcule. The body looks like a slipper. So it is having mouth. Mouth leads to gullet. The food along with the water, with the help of the cilia, it comes inside and the food will be digested inside. It has all the vacuoles and all to digest the food. It has dimorphic nucleus. One is macronucleus, other one is micronucleus. Macronucleus participates in asexual reproduction, micronucleus participates in sexual reproduction. It has two contractile vacuoles also, anterior contractile vacuole and posterior contractile vacuole. And paramecium divides by transverse binary fission. How does it divide? It divides by transverse binary fission. Coming to the last class, children, it is sporozoan. As we already discussed, in sporozoan there is no locomotory structure. There is no locomotor structure. That's why they are spore-like. And the example of sporozoan is the malarial parasite. What is it? Plasmodium, which causes malaria. Example for this one, Entamoeba histolytica, which causes amoebic dysentery. Example for flagellated protozoan, trypanosoma, which causes African sleeping sickness, which spreads by the bite of the CC fly. It spreads by the bite of CC fly. The third one is ciliated protozoan. Paramecium is an example. Here is the structure. Next one is a sporozoan, no locomotor structure, and the example here is a malarial parasite called Plasmodium vivax. There are four different types, right? Plasmodium vivax, Plasmodium ovale, Plasmodium malaria, Plasmodium falciparum. We can write any Plasmodium here, and this Plasmodium vivax, it causes malarial fever. It is a notorious parasite, we tell. It is a notorious parasite. It has a staggering effect. it has a staggering effect on human population. Means from that time till now, malaria has not been eradicated. It is there, it's suffering, people are suffering with this malaria. So then we call this a staggering effect. You know Alexander the Great, who defeated every king, right? So that's why we call Alexander the Great. He died with malaria only. Okay, so it has a staggering effect on human population. It's a notorious parasite. It killed Alexander also. And then the parasite is called Plasmodium vivax. Plasmodium, we can tell it doesn't have any locomotor structures. With this, we finish the second kingdom called Protista. Take a screenshot. We'll talk about the third kingdom called fungi. Now the third kingdom is fungi. Now what is a unique feature of fungi? Means they are completely heterotrophic. One complete kingdom which is having many number of organisms. All organisms are heterotrophic only. That too they are saprophytic. That's a unique habitat. Unique kingdom of heterotrophic organisms. And fungi are very much diversified. They show diversity in their morphology and their habitat. One type of fungal morphology is different from the other fungi. And they are diverse in their habitats also. Some are terrestrial, some are aquatic, some grow on cow dung, some grow on lignin, some grow on hair, like that. They are diversified in morphology and habitat. This point came in the NEET examination. Which kingdom has most diversity in habitat? Means in diversity in morphology means it's fungal kingdom only. The study of fungi is called mycology. And who is father of mycology? Michael is father of mycology. Study of fungi is called mycology. Now fungi are cosmopolitan. What do you mean by cosmopolitan? Air, water, soil, everywhere they are found. They prefer to grow in dark places, damp places and warm places. They prefer to grow in dark places, warm places and humid places. If the fungi is growing on wood, it's called lignophilous. If it grows on hair, keratophilous. If it grows on dung, coprophilous fungi it is. Usually fungi are multicellular except one member which is yeast, which is unicellular, which is kept under Ascomycetes. If you leave yeast, rest all fungi are multicellular only. The body of the fungus is long filamentous structures. The long filamentous structures, they are long, they are slender, they are extensions. These extensions are called hyphae. And these hyphae will make a network. The network of the hyphae is called mycelium. Because there are mycelial networks, study of fungi is called mycology. So the fungal body is filamentous. The filamentous are short or long? They are long, they are slender, they are thread-like. Those structures are called hyphae. And a network of this hyphae, branching of this hyphae, network of this hyphae is called mycelium. Now some fungi, the hyphae don't have septa, don't have partitions. Since they are not having partitions, it appears to be a common tube. In one tube, many nuclei will be there. That condition where septa are absent, many nuclei are seen is called cynocytic condition, multinucleated condition. We find it in the first class called Ascomycetes, whereas in the other classes, septa will be there. Since partitions are there, each cell will be having only one nucleus. Now, the cell wall of fungi is made up of chitin and polysaccharides. Fungal cell wall is made up of chitin and polysaccharides. Algal cell wall is made up of cellulose, galactans, mannans. Plant cell wall is made up of cellulose. Animal cell wall, they don't have cell wall, right? So in the same manner, what's the composition of fungal cell wall? Means it's made up of chitin and polysaccharides. As we discussed that fungi are showing heterotrophic mode of nutrition, means they grow on the dead and decomposing organic matter. They decay the dead and decomposing organic matter. Such type of fungi are called saprophytes. If the fungi are pathogenic on the living organisms, then they are called parasites. If they are on the dead body, saprophytic mode of nutrition. If they are on the living organisms, parasitic mode of nutrition. If they are making some symbiotic associations, algal fungal association is called lichen. And if the fungi are getting associated with the roots of the higher plants such as pinus, is called mycorrhizal associations. They make symbiotic associations. They are parasitic. They are saprophytic. Under symbiotic associations, fungi with algae is called lichens. Fungi with the roots of the higher plants is called mycorrhiza. Myco means fungi, rhiza means the roots of the higher plant. Take a screenshot. We'll talk about the reproduction of the fungi also. Now these are the characteristics of the kingdom fungi. Now when we talk about the reproduction, fungi will show vegetative reproduction also, they show asexual reproduction also, and they show sexual reproduction also. Let us see all the three types of reproduction exhibited by fungi, and we will see the three steps in the sexual reproduction also. Right? Now let's go. Vegetative reproduction is by which methods? Asexual reproduction is by how many types? And sexual reproduction is by how many types? Let us see. Fungi shows all the three types of reproductions. The first one is vegetative reproduction. Vegetative reproduction can be by fragmentation. The first method under vegetative reproduction is fragmentation. In fragmentation, this is the fungal hyphae. The fungal hyphae, which is having long filamentous like this, if the hyphae is broken, see this part of the fungal hyphae is broken. When the part breaks up, this establishes into a new individual. This is called fragmentation. It also shows binary fission. It also shows binary fission. In binary fission, the fungal hyphae, this is the fungal hyphae, will split into two daughter cells like this. This is one and the other cell. This is binary fission. So another type of vegetative reproduction is called budding. Budding is commonly seen in yeast. Yeast, which is an oval structure like this. Here a small projection will come, which is called as bud. Gradually the bud grows. Then the DNA, one copy of DNA, this is one copy of DNA which is getting into the bud. Now when the bud grows completely, when it gets its DNA, when it gets its all the contents, it detaches and then it becomes a daughter bud. So three types of vegetative reproduction, children. The first one is fragmentation, second one is binary fission, and the third one is budding. Now coming to how many types of asexual reproductions are exhibited by fungi. Asexual reproductions is by spore formation. It is by spore formation. Now let us see how many types of spores are there. So there are conidiospores, there are zoospores, and then we can tell they are sporangiospores, chlamydiospores. Different types of asexual spores are there. First, let us talk about the first type of spore, which is called zoospore. So zoospores means, zoo means motile, and these spores are formed inside, so they are endospores. They are formed inside zoosporangium. They're formed inside zoosporangium. Now this is zoosporangium. Okay, this is zoosporangium, and these are the zoospores. And these zoospores, since we are telling they are motile, these are the motile structures. Okay, and I draw it inside, so that means they are endospores. The second type of spore is called sporangiospore. The second type of spore is called sporangiospore, which is formed inside a place called sporangium. Which is formed inside sporangium. This is a vertical hyphae. Vertical hyphae will have sporangium. Inside the sporangium, all these are sporangiospores. All these are sporangiospores. Are they exospores or endospores, children? They are inside the structure, so they are endospores. But did I draw any motile structures? No, right? Means they are non-motile. So sporangiospores, if we have to tell, they are non-motile and they are inside sporangium, no? So that means they are also endospores. This is second type of asexual spore. Now coming to the third type, sexual reproduction, we'll see later. Third type of asexual spore is conidiospore. The third type of asexual spore is conidiospore, which is formed on a stalk called conidiophore. Now this is conidiophore. On this conidiophore, like a chain of conidia will be arranged. This type of structure we can see it in penicillium, right? So these are conidia, right? So they are exospores. They are outside. They are arranged in a chain-like manner, and they are non-motile. They are seen on a stalk called conidiophore. This is another type of asexual spore. Now, let us talk about the another type of asexual spore which is called as chlamydiophore. What is that? Chlamydiophore. Right? Another type of asexual spore is called chlamydospore. In chlamydospore, what happens? This is the hyphae. Now, under unfavorable conditions, the hyphae will collect all the reserve food material and it makes a spore-like structure and it becomes a thick wall structure. See, this cell has accumulated all the food and it has made a thick wall. Again, this cell also will accumulate and it will make a thick wall. So now these spores, thick wall spores, which have stored the reserve food material, it has stored the reserve food material and tide over unfavorable conditions, are called as chlamydiospores. They are called as chlamydiospores. So all these are different type of asexual spores. Vegetative reproduction is by fragmentation, binder fission and budding. Asexual reproduction is by zoospore, one type of spore. Zoospores are the common type of asexual spores in fungi. Second one is sporangiospores formed inside sporangium. Third one is conidia, which is formed on the conidiophore. Fourth one is chlamydiospores, which are thick walled resting spores which are formed during unfavorable conditions. Now when we have to talk about sexual reproduction, sexual reproduction is by different types of spores. Take a screenshot, we'll continue. Yeah. Now when we have to talk about sexual reproduction, what are the sexual spores which are formed? Sexual spores, we can name them as oospores, ascospores, basidiospores. These are the names what we give for sexual spores. Right? Now, vegetative reproduction, we told it is by fragmentation or by binary fission or by budding. And then we told asexual reproduction is by either zoospore or sporangiospore or conidia or conidiospore or chlamydospore. Aplonospores are also there. What is the difference between aplonospore and zoospore? Means zoospore is a motile spore, aplonospore is a non-motile spore. Now coming to sexual reproduction, when we have to talk about sexual reproduction in fungi, they form sexual spores. The sexual spores can be oospore, ascospore or basidiospore. Sexual spores can be oospore or ascospore or basidiospore. Oospore is formed on a fruiting body. Ascospore is also formed on a fruiting body. They are formed on special fruiting bodies. Ascospore is formed on a fruiting body called ascocarp. Basidiospore is formed on a fruiting body called basidiocarp. And oospore is formed on just we'll call it as a fruiting body. Now these are sexual spores which are formed on special fruiting bodies, right? And what are the 3 steps of sexual reproduction in fungi? In sexual reproduction, first what happens is, let us write down the 3 steps of sexual reproduction are: in the first step, the 2 hyphae, the 2 compatible hyphae, one is plus strain, other one is minus strain, they fuse with each other. Here they fused. When they are fusing, first their cytoplasms will fuse. So fusion of cytoplasm between the 2 cells. The first point is fusion of cytoplasm is called between the 2 cells, right? It is called plasmogamy. First cytoplasms will fuse, that is called plasmogamy. Then the nuclei also fuse, that is called karyogamy. Fusion of the nuclei will happen next. Fusion of the nuclei. What is it called? Karyogamy. Now, first cytoplasms will fuse, then the nuclei will fuse, karyogamy will happen, and the third step is after karyogamy happens, zygote will be formed. The zygote should undergo meiosis to form haploid spores. Spores on germination will form the hyphae again. So, fusion of the cytoplasm is called plasmogamy, fusion of nuclei is called karyogamy, followed by meiosis of zygote. Meiosis of zygote to form haploid spores. Zygotic meiosis will form to haploid spores. Now these spores on germination, the spores on germination will form the hyphae again, will form the body again. So these are the 3 steps of sexual reproduction. Once in NEET examination, they have asked to arrange these steps in order. Plasmogamy first, karyogamy next, zygotic meiosis next. So this order they asked once in NEET examination. Understood, children? So we have seen vegetative reproduction, asexual reproduction and sexual reproduction. Now on what basis we have divided fungi into 4 classes? Means on 3 points we have divided fungi into 4 classes. Now our first point is called as the morphology of the mycelium. The first point is on the basis of morphology of the mycelium. Means mycelium is longa, shorter, branchta, unbranched, septata, non-septata, multinucleata, single nucleata. Based on the morphology of the mycelium, based on the mode of the sexual spore formation. Second point, based on the mode of spore formation they told. In fact, which spore formation? Asexual spora, sexual spora means it is sexual spore formation. Based on morphology of the mycelium, based on spore formation and based on the fruiting body. Which type of fruiting body it is? Based on the fruiting body, we divide fungi into 4 classes. What are the 4 classes? The primitive class is called as Phycomycetes. The advanced classes are 2, Ascomycetes and Baisidiomycetes. The class which is not there now in existence is called as Deuteromycetes. So now we have divided fungi under 4 classes on the basis of 3 points. The first point is morphology, the second point is mode of spore formation, third one is fruiting body. Take a screenshot, we'll get into the classes of fungi. The first class is called as Phycomycetes. You know what is phycology? Study of algae. Means the first class appears like algal fungi. They appear like algae. They live in water only. So that's why the scientists have named the class as Phycomycetes. So the first class. What is that? Phycomycetes. Mysa means fungi. Phyco means algae. So Phycomycetes are commonly called as algal fungi. Why are they called as algal fungi? Like algae, they are aquatic forms only. We told on 3 points we have divided them into 4 classes. What's the first point? Morphology of the mycelium. Let us see how is the morphology of the mycelium. How is the morphology of mycelium? Now, the mycelium of Phycomycetes, it is unbranched, no branching, and it is aseptate also. Septa are also not there. Long hyphae will be there, but septa are not there. Since partitions are not there, many nuclei will suspend inside. Such a hyphae which is not having branches, which is not also having septa, is called as unbranched cynocytic, right? Multinucleated condition. Cynocytic hyphae we tell. Now, what is the second point we told? The second point we told, mode of spore formation. Mode of spore formation. Now, which type of asexual spore it will form? And which type of sexual spore it will form? Sexual spore is called oospore. The sexual spore formed by Phycomycetes is called as oospore. The asexual spores are zoospores. Asexual spores are zoospores. The sexual spore is called as oospore. On this basis, we have divided it into Phycomycetes. And then where are they found? Usually they are parasitic. They are obligate parasites on the plant. Phycomycetes members are obligate parasites on plant. Means they cause diseases for the plant. And what is the third one? Fruiting body we told, right? On the basis of the fruiting body, 3 points we have divided the fungi into 4 classes. The fruiting bodies are seen in sexual reproduction. The fruiting body is called as fruiting body only here. We did not give any specific name for the fruiting body of Phycomycetes. And asexual reproduction is by motile zoospores or by non-motile apranosaurs. Sexual reproduction is by oospores, right? Vested reproduction, fragmentation and all it will see. Now we have to see the examples. Means the first example is Mucor. The second example is Rhizopus. Rhizopus is commonly called as bread mold. And the third one is causing disease for the mustard. It causes white rust in mustard. It causes white spots on mustard. It is called albago. So the 3 examples, one is albago, which is parasitic on mustard. The second one is Rhizopus, which spoils the bread. And the first one is Mucor. These 3 are the examples of Phycomycetes. What did we study about Phycomycetes? Let's recall. We told Phycomycetes are algal fungi. Why are they called algal fungi? Because they're aquatic. They grow on decaying wood also. If they grow on decaying wood, we call them as coprophilous. Now I told they are obligate parasites on plants. Now on 3 points. The first point is morphology of the mycelium. Branched, unbranched, it is unbranched. Septa there are, septa are absenta, septate. Since septa are absent, many nuclei will be there, which is called cynocytic condition. Second point, on the mode of spore formation. A sexual spore is by motile zoospore or by non-motile apranospore. Sexual spore is by oospore. The fruiting body, it's not named, it's called as a fruiting body only. Examples are Mucor, Rhizopus and albago. Take a screenshot, we'll talk about the second class. Now the second class is called as Ascomycetes. The first class is Phycomycetes and the second class is Ascomycetes. Phycomycetes is called algal fungi. Then what is Ascomycetes called? Ascomycetes. Mycetes means fungi. Asco means sac. So that means Ascomycetes are commonly called as sac fungi, and Ascomycetes is the largest class among the 4. It is the largest class and it is the higher class also. Phycomycetes is a primitive class. Ascomycetes is a higher class. It is the largest class. It is called as sac fungi. Now they are called as sac fungi. They are multicellular except yeast, which is also there under Ascomycetes only. Only one unicellular fungus is there here. The unicellular fungus is yeast, whereas rest all fungi are multicellular only. Rest all are multicellular only. Let us talk about the 3 points. The first one is morphology of the mycelium. So they are branched. Branching is there. Septa is also there. Septate. Since septa are there, it is uninucleate. This is the first point. And the second point we told, the first point is the morphology of mycelium and second one is typospore formation. Typospore formation. Now, ascospores are formed. The sexual spore is ascospore, whereas the asexual spore, if we have to tell, the asexual spore is conidia. We have seen in Penicillium, this is conidiophore on the conidiophore. So these are conidia, chain-like non-motile conidia will come. So these are asexual spores which are called conidia. Sexual spore is called ascospore, which is formed inside a sac called ascus. Now inside one ascus, either 4 or 8 ascospores will be there. This is ascus. Now this ascospores are present inside ascus. They are non-motile, they are endospores, they are formed in a fruiting body called ascocarp. The third one we told, the fruiting body, right? The fruiting body is ascocarp. Now means we have to show an ascocarp here on which the ascus is there, inside which the ascospores are there. So these are the 3 points on which we have done the classification. Now if we have to see the examples, we have already seen the examples. The first example we wrote it is yeast, right? Now yeast, what it is, tell me. It is used in baking industry, so it is called as baker's yeast. It is used in alcohol industry also, so it is called as brewer's yeast also. This is one member. Next one is aspergillus. Next one is aspergillus. Aspergillus, what is it called? It's called laboratory weed, right? It is called, it freely grows inside the lab, so it's called lab weed. And this aspergillus will be there in the seeds, long stored seeds. It will be there. It produces a carcinogen called aflatoxin. Aspergillus releases aflatoxin. It releases aflatoxin, which is carcinogenic, which causes cancers. It is there in the stored grains. It will be there in the stored grains. So that's why we should not store grains too much long, because it gets infected with aspergillus. The first example for Ascomycetes is yeast. The second example is aspergillus. The third example is claviceps. Now claviceps, it is also parasitic. It causes ergot disease in rye. In rye plant, it will cause ergot disease. Now fourth example, penicillium. Penicillium, it produces antibiotic called penicillin. The fifth example is Neurospora. Neurospora is called Drosophila of the plant kingdom. It is widely used. The Neurospora is widely used in biochemical research and genetic research. That's why it's called Drosophila of plant kingdom. Neurospora is called Drosophila of plant kingdom. Now in plant, what they have taken, Mendel has conducted experiment in the pea plant. If we want to do fungi, we will take Neurospora and we will do biochemical research and genetic research. Apart from all these members, morels and truffles are also there, which add delicacies to the dishes. So fifth, sixth example and seventh example are morels and truffles, which add delicacies to the dishes. All these are examples of the second class Ascomycetes. First class Phycomycetes, primitive class. The second class Ascomycetes, largest class, higher class. Ascus means sac. Now they are unicellular. Yeast members are also there. Rest all are multicellular only. The nature of mycelium, it is branched children, it is septate, and only one nuclei will be there. And the spore formation, asexual spores are conidia, which are exospores, which are non-motile spores. Sexual spores are called ascospores. They are produced inside a sac called ascus. Ascus is present on a fruiting body called ascocarp. Examples are yeast, which is used in baking industry, so it's called baker's yeast. It's also used in alcohol industry, so it's called as brewer's yeast. The second example is aspergillus. It's called labweed. It produces carcinogen called aflatoxin seen in the stored grains. The third example is claviceps, which causes ergot disease in rye. The fourth example is penicillium, which produces antibiotic. Fifth example is Neurospora, which is widely used in biochemical and genetic research. The last examples are morels and truffles, which add delicacies they used in cooking. Now we'll discuss about the third class Baisidiomycetes. Take a screenshot. Now Baisidiomycetes are commonly called as mushrooms. They are commonly called as club fungi. So third class, what is it? Baisidiomycetes. Basidia means club, mysa means fungi. So I'm telling Baisidiomycetes are commonly called as club fungi. And which members are there? Mushrooms are here only. Bracket fungi. In bracket shape, they'll be there. Mushrooms, they're called bracket fungi. Some puffballs are also there here. right? So mushrooms are under Baisidiomycetes, bracket fungi are under Baisidiomycetes, puffballs are also under Baisidiomycetes. So these are some examples. Where do Baisidiomycetes grow? Where are mushrooms growing? They grow on wooden logs and all, right? So then what are the 3 points? This is also advanced class. This is also higher class only, advanced class only. Talking about the 3 points, the first one is the morphology of the mycelium we need to talk. Now the second one is the type of spore we need to talk. And the third one is the fruiting body we need to talk. The fruiting body we need to talk. Now coming to mycelium, the mycelium is branched, it is septate and it is uninucleate. The mycelium is branched. Like for Ascomycetes, it is branched, it is septate and it is uninucleate. Here the septa are dollipore septa, like bracket septa they will be there. These are the septa which is separating this cell from this cell. This septa is called as dollipore septa. So the mycelium is branched, means we can show a branch here. One branch is growing, one branch is going. So it is branched, it is having septa and it is uninucleate. Coming to the spore formation, the asexual spores are absent here. Asexual spores are absent. Sexual spore is basidiospore. The sexual spore is basidiospore formed on a special mycelium called basidium. Basidium is present on the fruiting body called basidiocarp. Basidium is present on a fruiting body called basidiocarp. Now if we have to see the basidiocarp, now this is basidiocarp, right? On the basidiocarp, now we have, this is basidiocarp. On the basidiocarp, this is basidium. Now this basidium undergoes meiosis to form 4 basidiospores. This is one basidiospore, second basidiospore, third basidiospore and fourth basidiospore. 4 basidiospores are formed. They are exospores, they are non-motile spores, they are formed on basidium special hyphae. Basidium is present on the fruiting body called basidiocarp. So then we talked all the 3 points. Morphology we talked, the mycelium is branched, yeah I showed you the branch. Septate, dollipore septa are there, only one nuclei will be there. And the spore formation, asexual spores are absent. Sexual spores are basidiospores formed on the basidium, present on the fruiting body called basidiocarp. Now we have to talk the examples of Baisidiomycetes. The examples of Baisidiomycetes, one is mushroom. Edible mushroom is called agaricus. Agaricus is edible mushroom. Poisonous mushroom is Amanita. Is poisonous mushroom. Now they also cause rust and smut. Right? Eusteligo causes rut, smut, and pecunia causes smut. Rust is caused by pecunia, smut is caused by eusteligo. Children, this point has been asked in the NEET examination. Rust is caused by pecunia, smut is caused by eusteligo, amanita is poisonous mushroom, agaricus is edible mushroom, and bracket fungi are also edible, puffballs are also edible. So all these are the members of Baisidiomycetes. One point in Ascomycetes and Baisidiomycetes, plasmogamy will not be immediately followed by karyogamy, because karyogamy is not happening immediately. Dikaryon stage will be there. This dikaryon stage will fuse, then it will form the zygote inside the fruiting body called basidiocarp. Right? Take a screenshot. Then we'll talk about the last class, fungus imperfecti, called Deuteromycetes. The last class is called as Deuteromycetes, which is also called fungus imperfecti. Why it is called fungus imperfecti means the perfect stage, the sexual reproduction stage is absent. Only vegetative reproduction and asexual reproduction is found in them. Now, the last class, class 4, which is called Deuteromycetes. Mysa means fungi, Deutero means imperfect fungi. So Deuteromycetes are commonly called as imperfect fungi. Why are they called as imperfect fungi? Means perfect stage, sexual reproduction stage is absent. Sexual reproduction stage is absent. So that's why it's called fungus imperfecti. Means vegetative reproduction we can find, asexual reproduction also we can find. Children, vegetative reproduction is by fragmentation. Asexual reproduction is by conidia. That chain like spores, exospores, non-motile spores are conidia. So coming to the morphology of the mycelium. It is branched, it is septate also, and it is uninucleate. And coming to the type of spores, as we told, sexual spores are absent. Whereas the asexual spore is conidia. And since sexual spore is absent, fruiting body is also absent. This was the point when they have done the classification, but now they even identified sexual reproduction in Deuteromycetes. When they identified sexual reproduction in Deuteromycetes, they removed the members from here and they placed them under Ascomycetes and Baisidiomycetes. Since the sexual reproduction is found, they have been moved off from Deuteromycetes and they are kept under Ascomycetes and Baisidiomycetes. What are the examples of Deuteromycetes? The first example, Alternaria. Second example, Coletotrichum. Okay, so Alternaria, Coletotrichum, and the third one is Trichoderma. These are the three examples of Deuteromycetes. Alternaria, what does it cause? Alternaria causes early blight in tomato. Alternaria is a pathogenic one. It causes early blight in tomatoes. Now, Coletotrichum causes red rot in sugarcane. Whereas Trichoderma causes ear rot in maize or in corn. So these are the three forms under Deuteromycetes. Alternaria causes early blight in tomatoes, Coletotrichum causes red rot in sugarcane, Trichoderma causes ear rot in maize. And why it is called fungus imperfecti? Because at the time when they have done the classification, sexual reproduction is absent. Then which manner they reproduce? Vegetative reproduction by fragmentation, asexual reproduction by conidia. So take a screenshot. We have finished fungi. Then we will start with the plant kingdom and the animal kingdom. So we finished Monera, Protista, fungi. Then the last two kingdoms are, in Whittaker's 5-kingdom classification, are Plantae and Animalia. So I already took the points about Plantae and Animalia. Let us see quickly on the board. So the 4th kingdom is plant kingdom and the 5th kingdom is animal kingdom. Now, only 1st kingdom includes unicellular prokaryotes. Now the 2nd kingdom, Protista, 3rd kingdom, fungi, Plantae and Animalia, all are eukaryotes only. But Protista is unicellular eukaryotic kingdom, whereas fungi, Plantae, Animalia are multicellular eukaryotic kingdoms. So the first characteristic of plant is they are eukaryotic, and plants are producers, means they contain chlorophyll, they do photosynthesis. They are eukaryotic, they have chlorophyll containing, they are multicellular forms, right? They do photosynthesis, they can trap the sunlight energy and convert the light energy into chemical energy which is used for synthesis of the sugar, right? So few are partially heterotrophic. So not all plants are producers. There are some plants which are partially heterotrophic, means they can prepare their food but they grow in nitrogen deficient soils. To get the nitrogen, they'll be killing insects. Then we are talking about insectivorous plants like Nepenthes, which is called pitcher plant, Dionia, which is called as Venus flytrap, Drosera, which is called sundew plant, Utricularia, which is bladderwort. So all these are the examples of insectivorous plants. They manufacture their food, they're green, they do photosynthesis, but since they are growing in nitrogen deficient areas, they kill the insect and from the insect's body they take the nitrogen. So some are partially heterotrophic, which are called insectivorous plants. The examples are Venus flytrap, it's called Dionia. Venus flytrap, the leaf will be bifid like this and it will be very colorful. So the insect gets attracted to the color, and when it comes, it traps the insect. It closes and it kills the insect. Whereas bladderwort Utricularia is an aquatic plant. It will be there in the water, suspended inside the water. It has bladder-like structure. The insect, when it swims and comes, it opens and it takes the insect inside the bladder. So these two are partial heterotrophic. But there are some plants like root parasites, stem parasites, they cannot prepare their food. So they don't have green color pigment in them, and they depend on the host plant. So for example of parasites are, in NCERT they have given Cuscuta plant, dodder plant, Cuscuta plant. It is a complete stem parasite. Means it cannot prepare the food, nor it can absorb the water from the host plant. It takes water also, it takes food also. So I'm telling it's a complete parasitic, right? And plant cells have cell wall. The cell wall is made up of cellulose. So cellulosic cell wall they have. And the plant kingdom is divided into 5 groups: algae, bryophytes, pteridophytes, gymnosperms, angiosperms. Algae are primitive aquatic plants. Gymnosperms, angiosperms are advanced plants. Bryophyte, pteridophytes, they come under the intermediate categories. So we have written that the plant kingdom has 5 groups: algae, which are called thallophytes; bryophytes, which are called amphibians of plant kingdom; pteridophytes are the first vascular plants; gymnosperms, naked seeded plants; angiosperms are flowering plants. So these are the 5 groups which we are going to see in detail in the chapter 3 plant kingdom. Now we told plant has alternation of generations, means it has a life cycle which alternates: gametophytic stage, sporophytic stage. Gametophytic stage means gamete making stage. Sporophytic stage means spore making stage. Sporophytic stage is diploid by nature. Gametophytic stage is haploid by nature. From gametophyte it will come to sporophyte. From the sporophyte it will go to gametophyte. This is alternation of generations. And gametophyte does not resemble sporophyte in appearance, so means they are heteromorphic. The life cycles are heteromorphic. They show alternations in the life cycles, means they have 2 stages: gametophytic stage and the sporophytic stage. Gametophytic means gamete making stage, and gametophytic stage is haploid. Sporophytic stage means spore forming stage, and they are diploid. Since they alter with each other, it's called alternation of life cycles. And since they don't resemble in the structure, it's called heteromorphic. Now they are variable. The gametophytic stage in one group of plant is more, the sporophytic stage is restricted. In other group of plants, sporophytic stage is more, the gametophytic stage is restricted. In another group of plant, both are almost equal. So basing on the duration of the gametophytic stage and the sporophytic stage, basing on which phase is dominant and which phase is dependent, we can divide the plant life cycles into haplontic life cycle, diplontic life cycle, haplodiplontic life cycle. Algae are examples of haplontic life cycle where gametophyte stage is dominant. Gymnosperms and angiosperms are examples of diplontic life cycle where the sporophyte stage is dominant. Whereas coming to bryophytes and pteridophytes, they have an intermediate life cycle where gametophytic stage and sporophytic stage both will be there almost at equal proportion. So children, this is about the characteristics of the plant kingdom. As I told you, we will detail plant kingdom in detail in the next chapter, chapter 3. So if we have to just summarize about the 4th kingdom, Plantae, in R.H. Whittaker's 5-kingdom classification, we are telling plant kingdom includes multicellular, chlorophyll bearing, and they are eukaryotic organisms. They contain chlorophyll, they do photosynthesis, they are producers. There are partially photosynthetic organisms are there. They grow in nitrogen deficient soils. For nitrogen, they kill the insects. They are called insectivorous plants. 2 examples in NCERT which they have mentioned are Venus flytrap, common name is called Dionia; bladderwort, which is called as Utricularia, they have given. And under parasitic plant in NCERT, they have taken the example of a dodder plant which is called Cuscuta. It's a complete parasite. It depends on the host for food also, water also. There are 5 groups of plants which come under plant kingdom. Primitive are algae, then comes bryophytes, pteridophytes, gymnosperms and angiosperms. Plant shows alternation of generations and they are heteromorphic. Haplontic life cycle is expected majority of the algae. Diplontic life cycle is expected of gymnosperms and angiosperms. Bryophytes and pteridophytes shows intermediate life cycle. I think we have done with plant kingdom. Now let's move on to the last kingdom, that is animal kingdom. So what are the differences from plant kingdom to animal kingdom? They are photosynthetic, they are autotrophic, they are heterotrophic. So they have cell wall, they don't have cell wall. Plants cannot move, animals will move. Plant has indefinite growth. It keeps on growing because of the presence of meristematic tissue. Animals have a definite growth. right? So let us see the differences. Animal cells will have centriole, plant cells will not have centriole. Plant cell will have chloroplast, animal cell will not have chloroplast. So these are the differences. The reserve food material in plant is starch, the reserve food material animal is glycogen, right? Let us see the differences. Now coming to the last kingdom, animal kingdom. Animal kingdom also includes eukaryotic only, multicellular, and they are motile, right? They are moving organisms. Now they are heterotrophic. These are autotrophic, they are heterotrophic mode of nutrition. So the animals which directly depend on plants for food are called herbivores, grass eating animals. The animals which indirectly depend on the plants for food are called carnivores, right? So they digest the food in the internal cavity, means they have digestive system, alimentary canal, there they digest the food and they store the food either in the form of glycogen or in the form of oil. The reserve food material for animal is glycogen, the reserve food material for plant is starch. Now as I told, animals have a definite growth pattern. After certain age will not grow, my height is fixed. So they have a definite growth pattern and they have complex sensory structures. So we can sense our surroundings. They have neuromuscular structures also. They can move with the help of muscles and their response is because of the neural signals. They are capable of locomotion. And plant kingdom is divided to 5 groups, whereas animal kingdom is divided to 2 phyla, chordates and non-chordates. Now they exhibit sexual reproduction. Sexual reproduction means copulation, mating, where the gametes will be fused to make a zygote. Zygote develops into embryo, embryo develops into an individual. These are the characteristics of the animal kingdom. If you have to just summarize the animal kingdom, so we'll tell they are eukaryotic, they are multicellular, they are motile organisms. They have heterotrophic mode of nutrition. They cannot prepare their food. Cell wall is absolutely absent and they are directly dependent on the plants or indirectly dependent on the plants. The animals which directly depend on the plants are called grass eating animals, herbivores. The animals which indirectly depend on the plant are called carnivores, right? And they digest the food in the digestive system and store the food in the form of starch or glycogen or oil. No cell wall, they have a definite growth pattern. After that they don't grow, whereas plant keeps on growing. Higher animals, they have sensory structures, they have neuromuscular structures also. Animal kingdom is divided into 2 groups, chordata and non-chordata. And sexual reproduction is by copulation, it's by mating, where the gametes will fuse to make the zygote. Zygote undergoes embryogenesis. And detailed description about animal kingdom we are going to see in the chapter 4 called Animal Kingdom. So take a screenshot, we will continue. Alright, so according to RH Whittaker 5 kingdom classification, we have seen all the 5 kingdoms, right children? Monera, Protista, Fungi, Plantae, Animalia. Now at the ending of the chapter, the author is telling that there are some organisms which were not placed by Whittaker, means they are not meeting the criteria, the 5 points of criteria what he has taken: body organization, cellular organization, mode of nutrition, phylogenetic history. So there are some organisms which are not meeting the criteria since they are acellular. Cell is only not there, then where he can keep? Prokaryotica, eukaryotica he cannot keep. So in Whittaker's 5 kingdom classification, there is no mention about certain acellular organisms like viruses, viroids and lichens. Okay, so that also we will discuss and then we can end the chapter. So we are writing that in Whittaker's 5 kingdom classification. There is no mention about, he couldn't mention about certain acellular structures, right? He did not mention about certain acellular structures. What are those acellular structures? Means one, viruses. The second one he called they are viroids. And the third ones are lichens. Lichens are associations. They have prokaryotic and eukaryotic organism within. So lichens are associations. Associations he couldn't find a place to keep. And viruses and viroids are acellular organisms. So that also he couldn't find any place to keep. So that's why he couldn't keep them. Now let us discuss about viruses. We can't ignore viruses. Viruses are pathogenic. They're disease causing. We need to understand the physiology of viruses also. So let us start with viruses. Right, So now what are viruses? Are they cellular or non-cellular? They don't meet all the characteristics of a cell, so we better to call it as non-cellular only. They are living or non-living. So they are non-living only since there is no metabolism, but they are infectious. We can tell viruses are acellular. They are infectious and they are non-living. Right? They are non-living. So we can instead tell that viruses acts as a connecting link between living organisms and the non-living things. We told Euglena acts as a connecting link between plant and animal, since it has plant characters also, animal characters also. In the same manner, we can also tell that viruses act as connecting links between non-living and living things. We can also call viruses as connecting links in non-living things and living things. Means I am telling that viruses will possess certain non-living characters also, and viruses will possess living characters also. Now let us see what are the non-living characters viruses will possess and what are the living characters viruses will possess. Now virus, if it is outside the body, outside the host, then we tell it is crystalline, it is inert solid substance. It's not like cell, it's just crystalline as a chemical. So non-living character is outside the cell. Outside the cell, they are inert. They are crystalline structures, so which states that it is non-living only, and they don't have their own metabolism. Metabolism is a defining character. Those organisms which don't have metabolism are non-living only, so they don't have metabolism, no metabolism, and they don't meet all the characteristics of a cell, so we can call they are acellular. They are acellular. That also we can tell. And outside the host, they cannot multiply also. They cannot multiply. Means reproduction, if they are reproducing, that means it's a living characteristic. They cannot multiply, they cannot reproduce. So it's a non-living character. They cannot multiply. All these are non-living characters. Coming to living characters, we can tell they are infectious. We can tell first one, they are obligate parasites, compulsory parasites. They are obligate parasites. That's the first point. And second point, we can tell they are infectious. They cause diseases, right? They are infectious. They have DNA or RNA as a genetic material. They have DNA or RNA as genetic material. Since they are having DNA or RNA as genetic material, they can show mutations. They exhibit mutations. And using the host machinery, it can multiply and it can produce its daughter generations. Virions it can produce. Infectious viruses it can produce. So that is also a living character. So we can tell in host, virus multiplies and produces. It multiplies and produces virions. In host, the virus multiplies and it produces virions. All these are the living characters. So we are telling it's an obligate parasite. It stays inside the host. Means it's an intracellular parasite. It's an obligate parasite, and since it is staying inside the host cell, we can tell it is intracellular parasite, and it is infectious. It causes diseases. It has DNA or RNA as the genetic material, and that genetic material exhibits mutations. Inside the host, it multiplies and it produces its progeny. All these are living characteristics, right? So we finished 5 kingdoms. Let us summarize what is there on the board. We finished 5 kingdoms, and we are talking about R.H. Whittaker couldn't place certain organisms in all these 5 kingdoms. What are they? They are viruses, they are viroids, and they are lichens. Prions are also there. So when we talk about viruses, they are acellular, they are infectious, they are non-living. In fact, we can tell viruses are connecting links between living and non-living organisms. These are the non-living characteristics of viruses, and these are the living characteristics of viruses. Take a screenshot, we'll continue. Now, when we have to talk about the size of viruses, viruses are very small, so we cannot see it under light microscope. Viruses are visible only under electron microscope. So we can tell viruses are ultra microscopic since they are seen only under electron microscope. Okay. Now, what is the smallest virus? And what's the name of a biggest virus? Foot and mouth disease virus is the biggest virus. You know who coined the term virus? What is the meaning of the word virus? Virus means venom, poison. And a scientist called Louis Pasteur, he named the term called virus. He coined the term called viruses. So we are continuing with viruses only. When we have to talk about the size of the virus, we are telling they are ultra microscopic. So size, we are telling viruses are ultra microscopic. Means they are seen only under electron microscope. They are seen only under electron microscope. So if we have to take this largest virus name and if we have to talk about the smallest virus name, let us see that fast. The largest virus is pox virus. Right? So pox virus is an example of largest virus, and vaccinia virus is also example of largest virus. Vaccinia virus are examples of largest virus. And a small virus, we can tell it is polio virus, it is small virus, and foot and mouth disease virus. In cattle it causes a disease called foot and mouth disease virus. Right? So foot and mouth disease virus and polio viruses are example of smallest viruses. Pox virus and vaccinia virus are example of largest viruses. And what is the meaning of the virus? So virus means venom, means poisonous fluid. It is poisonous fluid. Bacteria, we can tell good bacteria there, bad bacteria there, but we cannot tell there's a good virus. Means virus meaning itself is poisonous fluid. And Louis Pasteur is a person who coined the term virus. So who coined the term virus? Louis Pasteur. He coined the term virus. He coined the term virus. And the study of virus is called virology. What is virology? Virology is the study of viruses. Now, who are those scientists who made discoveries related to viruses? If you see, first scientist comes as D.J. Ivanovsky. D.J. Ivanovsky. What did he tell? He tells that viruses are filterable. Ivanovsky tells that viruses are filterable. So he has seen a tobacco plant is suffering with tobacco mosaic virus. He tried to pass it. He tried to pass that filtrate through the bacterial filters. But since viruses are ultra microscopic, so they can pass through the bacterial filters, they came down. Then basing on that, Bijernik told that viruses are filterable. In which year he told this? He told it in the year 1872. He told viruses are filterable. Then came Bijernik in the year 1898. In the year 1898, now what did Bijernik do? So he has taken the liquid from the infected plants, he filtered it and he took the liquid of the infected plants, and when he sprayed it on the healthy plants also got the disease. Then he told this is fluid which is having that infectious agent, it is spreading. Then he used a phrase called contagium vivum fluidum. The fluid is contagious, it can transmit disease from one plant to another plant, he told. So Bijernik in year 1898, he used a phrase called contagium vivum fluidum. He told the fluid is contagious. In which year? In the year 1898. What's the name of the scientist? Bijernik. And who isolated it and who crystallized it? Who gave the structure of the tobacco mosaic virus? Means it is Stanley in the year 1935. In the year 1935, the name of the scientist is Stanley. Now what did he do? He isolated tobacco mosaic virus and he crystallized and he gave the shape of the tobacco mosaic virus also. Stanley in 1935, he isolated tobacco mosaic virus. He even crystallized tobacco mosaic virus. He even gave the, if you crystallize it, you can give the shape. He also gave the shape of tobacco mosaic virus. He told tobacco mosaic virus is rod-shaped structure. It is rod-shaped structure, he told. So this is a contribution of Stanley. Next, Adolf Meyer. He identified the infectious nature of tobacco mosaic virus. What is the name? Adolf Meyer. What did he do? He identified the infectious nature of tobacco mosaic viruses. So most of the discoveries, initial discoveries, are based on plant viruses only, that is tobacco mosaic virus. Now, who coined the term virus? The virus word was coined by Louis Pasteur. Louis Pasteur is a scientist who coined the term virus. The meaning of virus is venom or poisonous fluid. Then came Ivanovsky. He told viruses are filterable. He passed it through the bacterial filters and virus came down because they are very small. Then came Beijerinck. Beijerinck used a phrase called contagium vivum fluidum. The fluid is contagious, he told. Then came Stanley. He isolated tobacco mosaic virus. He crystallized the virus also and he told the shape of the tobacco mosaic virus. It is rod shaped. Then Adolf Meyer identified the infectious nature of tobacco mosaic virus. And the viruses which infect plants, are called as phytophages. The viruses which infect animals are called zoophages. The viruses which infect bacteria are called bacteriophages. The viruses which infect plant are called phytophages, which infects animals are called zoophages, which infect bacteria are called bacteriophages. The viruses which infect cyanobacteria are called cyanophages. The viruses which infect cyanobacteria, what are they called? They are called cyanophages. They are called cyanophages. Now, who is the scientist who identified bacteria infecting virus? Means D. Herrell. I write it down here. D. Herrell, he identified bacteriophage. Now, what is bacteriophage? The virus which infects bacteria is called bacteriophage and D. Herrell identified bacteriophage. Now, the disease, sexually transmitted disease, what is that disease? It is AIDS. AIDS is caused by a virus called HIV virus. The structure of HIV virus was given by Gallo and Montagnier. The structure of HIV was given by Gallo and Montagnier. So this is a scientific background about viruses. Take a screenshot, we'll continue. Right. So when we have to talk about the composition of the viruses, viruses are called as nucleoproteins. Means they have nucleic acid and they have a protein coat also. If the lectures are informative, comment them in the comment box. If you need any requirement, that also you can post it in the comment box. I think this will be an ultimate platform for you to crack the NEET examinations. Let's talk about the composition of viruses. Right? So, viruses are called nucleoproteins. Means they have nucleic acid and they have protein coat. The protein coat, they don't have cell wall, okay? They have a protein coat. The protein coat is called capsid. And the capsid is made up of many monomers. The monomers are called capsomeres. What are the monomers of the capsid called? Capsomeres. And then it has, the core is a nucleic acid. The core is a nucleic acid. Nucleic acid is wrapped by a protein coat. The nucleic acid can be either DNA or it can be RNA. Okay. So the viruses which are having DNA are called as deoxyriboviruses. And the viruses which are having RNA are called as riboviruses. The viruses which are having DNA are called as deoxyriboviruses, and the viruses which have RNA are called as riboviruses. Generally, plant viruses have RNA as a genetic material, animal viruses will have DNA as a genetic material. So it is having an outer coat called capsid. Capsid is proteinaceous in nature and it is made up of monomers. Each monomer is called as capsomere. Now, in some viruses, external to the capsid, covering the capsid, one more layer will be there, which is called as external to capsid. It has envelope. Now, those viruses which are having envelope covering the capsid are called as enveloped virus. HIV is an example of enveloped virus. Coronavirus is also example of enveloped viruses. So, in some viruses, external to the capsid, they have envelope and they are called enveloped viruses. HIV is an example of enveloped virus. And then we are telling, so plant viruses, they have, what do they have? Plant viruses have RNA as genetic material, whereas animal viruses, mostly I am telling, animal viruses have DNA as genetic material, but exceptions are there. Now, if we have to talk about cauliflower mosaic virus and dahlia mosaic virus. If we have to talk about cauliflower mosaic virus and dahlia mosaic virus, they contain double-stranded DNA as a genetic material. They contain double-stranded DNA as a genetic material. Even though they are plant viruses, they are having double-stranded DNA as a genetic material. Now, usually animal viruses have DNA as a genetic material. Now, if you take HIV, which is an animal virus, HIV has single-stranded RNA as a genetic material. HIV will have single-stranded RNA as genetic material. Now, bacteriophages, if we take, means the viruses which infect bacteria are called bacteriophages. Bacteriophages, usually they have double-stranded DNA as genetic material. So, plant viruses have RNA as genetic material in most of the cases. Animal viruses will have DNA as genetic material, but the exception is CMV, cauliflower mosaic virus and dahlia mosaic virus. Even though they are plant viruses, they have double-stranded DNA as genetic material. Now, animal viruses should have DNA, whereas HIV virus have RNA as genetic material. Bacteriophages, the viruses which infect bacteria are called bacteriophages. They should have double-stranded DNA as a genetic material. But when we take about a bacteriophage called PX174 and M13 and Q14 coliphases. Now, these are some examples. What are they? One is PX174 we can tell. Next one is M14 and Q13. M14 and Q13 coliphases, they contain single-stranded DNA as a genetic material. Bacteriophages like PX174, M14 and Q13 bacteriophages, coliphases will have single-stranded DNA as a genetic material. Take a screenshot. Now, usually RNA, if it's a genetic material, it will be single-stranded RNA. Now, name any virus which has double-stranded RNA as a genetic material. Plant viruses, we are telling they have RNA as a genetic material, but that RNA is single-stranded RNA. There are some examples like wound tumor virus, which has double-stranded RNA as a genetic material. All exceptions. Next one is wound tumor virus, Rio virus. What do they have? Double-stranded RNA as a genetic material. They have double-stranded RNA as a genetic material. Now, viruses means poison, poisonous fluid. They are infectious. They cause diseases. What are the diseases? AIDS is caused by HIV virus only. The next one, mumps is caused by mumps virus. Smallpox, smallpox virus. Influenza, influenza virus. So, all these are the animal diseases caused by viruses. If you have to list down some animal diseases caused by viruses, right? Mumps, measles, then next one influenza, then we are telling common cold, AIDS, right, and herpes disease. Okay, so hepatitis, N number of diseases are there. COVID, all these are examples of the viruses which cause animal diseases. Now if you have to take about plant diseases, now they cause mosaic disease, leaf fall disease, right? And what are the symptoms the plant will exhibit if it is having a viral disease? What are the symptoms the plant will exhibit? Means leaf rolling will happen, leaf curling will happen, mosaic patches will come, yellowing of the leaves. All these are the symptoms if we find in the plant, so we can make out that the plant is suffering with a viral disease. What are the symptoms the plant will exhibit when it's having a viral disease? The first one is mosaic patches. Mosaic patches on the leaf is one symptom. Rolling of the leaf rolls off is another symptom. Rolling or curling, we can tell. Curling of the leaf is another symptom. Then next one we are telling yellowing of the veins. Yellowing of the veins. Stunted growth. Yellowing of the veins, stunted growth, vein clearing we tell. Yellowing of the veins is called vein clearing. All these are the symptoms the plant will show when it is suffering from a viral disease. So, and if we have to see the structure of tobacco mosaic virus which is there in NCERT, they are showing us these are capsomeres. Right, these capsomeres are arranged, and when we see, it will be like a rod only. So what I am drawing are the capsomeres of the virus, and inside tobacco mosaic virus, it has single-stranded RNA as a genetic material. Now the single-stranded RNA is having helical symmetry, and we can see these, right, helically coiled single-stranded RNA it is. So this is single-stranded RNA. This is a shape of tobacco mosaic virus. This is single-stranded RNA, and these are capsomeres. Now if you have to see the structure of bacteriophage. Now bacteriophage has a head which is having icosahedral symmetry. It has a head which is having icosahedral symmetry. So, and then it has a collar here, and then it has a body. At the end, it has a hexagonal plate. To the hexagonal plate, it has tail fibers at each corner, and it has basal pins to pin the bacterial surface, and these are the sheets which make the body. So this is the shape of a bacteriophage. So in this, we call this as a head region which shows icosahedral symmetry. This is collar, and this is a body, or we can call it as a tail sheath, and this is a hexagonal basal plate, and these are basal pins, and these are tail fibers. The typical shape of a bacteriophage is tadpole shaped structure. It is tadpole shaped structure, and the name bacteriophage was coined by D. Harrell, right? It infects bacteria, so that's why it's called bacteriophage. Now this is the information about viruses. Take a screenshot, then we'll talk about viroids, prions and lichens also. Yeah. Now what are viruses? Viruses are having genetic material inside. It can have either DNA or RNA as a genetic material, but it will not have DNA and RNA both. This point came in NEET examination once. Viruses have either DNA or RNA as a genetic material, but they don't have both inside the body. Right? So what are basically viruses? Viruses means they have a protein coat outside, and they have genetic material inside. They have genetic material inside. Now if both are there, then it is called as virus. There are certain structures which are called Viroids. T.O. Diner discovered viroids. T.O. Diner discovered viroids. Now, how are viroids differ from viruses? Means the viroids are naked particles. They don't have protein coat. So, viroids, they lack the protein capsid. They don't have protein capsid. That means they are naked. Then what do they have? They have small, low molecular weight single-stranded RNA as the genetic material. They have only single-stranded RNA which is not wrapped by a protein coat. Then we call them as viroids. What are the diseases caused by viroids? Means potato spindle tuber disease. So a viroid causes what disease? It causes potato spindle tuber disease. Okay, so the pictures related to this chapter, I'll post the link in the description that you can see. You can click and you can see the pictures. If we visualize them, the content will be memorized for us easily. So I'll post the pictures of bacteriophage, I'll post the pictures of potato spindle tuber disease, I'll post the pictures for vein clearing, stunted growth, rolling of the leaves. I'll post, just watch the pictures in the description box. I'll share you the link. So it causes potato spindle tuber disease. So this is information about viroids. Viroids are discovered by T.O. Diner. And what are viroids? Viroids are naked because they don't have a protein coat. Then what do they have? They just have a small low molecular weight RNA as the genetic material. This also has been asked in the NEET examinations. Viroids are also infectious. They are also pathogenic. They cause diseases. One such disease is potato spindle tuber disease. Now another alteration is prion. Now prions were discovered by Prasanna. Prions were discovered by Prasanna. See, viruses means it has protein also, it has genetic material also. Then when you talk about viroid, in viroid protein is absent, only genetic material is present. In opposite way, if you talk about prion, in prion protein will be there, genetic material is absent. So I think you understood. So prions were discovered by Prasanna. What are prions? Highly folded infectious proteins. That's it. Prions means they are highly coiled, excuse me, they are highly coiled infectious protein particles. They are highly coiled infectious protein particles which are identified by Prasanna. So they cause diseases. What diseases prions will cause? So it causes CJD disease in animals and it causes BSE in animals. Now what do you mean by BSE? Bovine spongiform encephalopathy. BSE means it is bovine spongiform brain infection encephalopathy, which is equivalent to CJD disease in humans, like CR-Jacobs disease in humans. So this is a disease caused by prions. So viruses, viroids, prions were not included by Whitaker in his Phycomycetes classification. And if we come to associations, lichens are there. Lichens are also not included in his associations in the Phycomycetes classification. What are lichens? Take a screenshot. We'll talk about lichens also. Now the last topic in this chapter is with lichens. Now lichens are symbiotic associations. They are associations between algae and fungi. Where do we find them? We find them on the woods. And lichens are very good pollution indicators. If pollution is there, lichens will not grow. If lichens are growing in an area, means it's an unpolluted area. So let's talk about lichens also. What are lichens? They are symbiotic associations between algae and fungi. Algae and fungi join to make lichens. The algal component is called phycobiont. The fungal component is called mycobiont. Study of algae is phycology. So the algal component is called phycobiont. The fungal component is called mycobiont. Now what algae will do? Algae will prepare food. Algae will prepare food and supply it to fungi. What fungi will do? Fungi will absorb water and it gives shelter to the algae. It gives shelter to algae, and fungi participate in reproduction also. It participates in reproduction also. And which type of algae will be involved in lichen associations? Means they belong to blue-green algae and green algae. So which phycobiont? It is either blue-green algae, Cyanobacteria, either blue-green algae or green algae are phycobiont members. Under mycobiont, they are either Ascomycetes members or Baisidiomycetes members. They are either Ascomycetes members which will make associations or Baisidiomycetes members which make associations. And then we are telling lichens are very good pollution indicators. They are very good pollution indicators. Now they indicate air pollution. They indicate air pollution. They don't grow in polluted air. Do not grow in polluted air. They denote in air which pollution? They denote SO2 pollution, sulfur dioxide pollution. They denote sulfur dioxide pollution. Children, all this is information about the organisms, the acellular organisms which are not placed by R.H. Whittaker in his 5 kingdom classification. So hope the lecture is informative up to the standard of the NEET. So the pictures related to this chapter, I'll share the link in the description box. You can open and you can see the pictures also. So we'll meet soon with the next lecture. Thank you.